Okay. Hello, everyone. Welcome to the 2024 edition of the Spinal Cord Toolbox course. My name is Julien Cohen-Haddad. I apologize for my voice. I happen to have a cold. I think it's not the first time this happens during SCT course. That's a unfortunate coincidence. But anyway, we'll see each other for 2 days. So we started today at 10 a.m. Eastern Standard Time, and then we'll finish at 2 p.m. So we'll be together for a few hours. And let me just pull the agenda for today. I'm gonna share my screen. Okay, so this is the agenda that you can find on the website. So we're gonna start with little introduction, and then we're gonna start right away with the course. We're gonna have a very quick lunch break, so I hope that you have your lunch prepared because you only have half an hour. Then we'll continue and then finish at 2 p.m. We decided to go with this very narrow time to accommodate different time zones before us and after us. And tomorrow we have the similar schedule, starting at 10 and then finishing at 2 p.m. EST. Let me pull the slides. Okay, so what I'll do is you can see that there's a little speaker note. This is for me and for the people presenting. First off, I'll be the one speaking most, but in the end, SCT is an open source project that started about 10 years ago. And it's really a teamwork. And the current team, the core developers, include Joshua Newton and Mathieu Gay-Paquet. So, big recognition for their involvement and hard efforts. And a lot of content has also been developed over the years by the different students, interns, some of which will be presenting today, including Sandrine Bédard, Yann Valochek, Naga Kartik, and Nathan Molinier. This course is extremely informal. The goal is really for you to get familiar with SCT. Some of you already are, but nevertheless, we are used to heterogeneous crowds. So if you have specific technical questions, feel free to ask. If you have very basic question, feel free to ask as well. There is no stupid question at all. We're basically here to help. And you can ask your question on the chat. You can also interrupt me anytime. Raise your hands, by clicking the hand raising button, and so I will see you. Some of the people in the past were, especially when we had courses like on site, people would usually print these slides, and so one way to print them so that you also have the speaker note is explained here, basically. So we have a very packed agenda, and this agenda is organized in 2 days. We're gonna try to finish after this, but depending on how many questions we have, how many time we need to spend, we might be longer there or like faster depending on the pace. But it follows the natural evolution of a pipeline, starting with very basic the basics of spinal cord segmentation. That is really the start for many of the subsequent analysis, and then we'll go with more complex types of analysis. finishing with DMRI, functional MRI, other types of features, and so on. All right, so very brief recap of what quantitative MRI is. It's essentially an umbrella term for the way we can push special buttons on the MR scanner to expand our visibility of the tissue microstructure and function. A typical clinical MRI scans like T2-weighted or T2 flare, T1-weighted proton density. They give you a lot of insights, visually speaking, the presence of a lesion, for example. But if you want to go deeper, like quantifying the number of the lesions, quantifying their individual size, quantifying subtle spinal cord atrophy of less than 2%, all that requires advanced image analysis tools that essentially SCT provides. So this, you can see SCT as an equivalent to the tools that have been on the horizon for 2-3 decades now, including FSL, FreeSurfer, SPM. And SCT is the equivalent but for the spinal cord, because many of the features that FSL, SPM, and so on provide don't adapt or don't apply to the spinal cord. We don't want to replace some of the features that can be used with FSL or other libraries, which is why I like to call SCT more of a pre-processing pipeline as opposed to a holistic pipeline. To be more, to give you an example, for people doing functional MRI, they can use SCT for some of the pre-processing that is very spinal cord specific, like segmenting the spinal cord, motion correction, registration to a spinal cord template. But then for higher level second type analysis for statistical purpose, then you can use many other tools that are not agnostic or like that are agnostic to the organ of interest. We'll talk about this a bit later. Any questions so far? I will try to pull my chat box so that I can see if there are some questions on the fly. Yeah. Okay. Cool. Okay. So we try to publish most of the methods, if not all the methods that go into SCT, to basically show that they are validated, and to provide a comprehensive and transparent methods of all the features that are implemented in SCT. And the list is evolving as the students evolve in the lab. As you can see, there are some recent studies that came out. And then the number of citation is also growing. I see more and more fMRI applications, and then there is also continuous applications of SCT in various diseases, including MS, ALS. If you're interested in having an overview of the applications, you can click on this link, and then you have so we keep track of the citations of SCT and also what are the tools that are used and the pathologies that are involved. So we find this pretty informative, and it's also important for asking for funds to sustain this software ecosystem. It's getting pretty old now, almost 10 years, but it was one of the first study that was done not by us, but by Marios Giannakas at UCL. They compared SCT with, at that time, a reference for spinal cord cross-sectional area measures and with an application in MS. And basically showed that this fully automated method is provides similar results in terms of reproducibility, and that could be extended to very large databases. And then a more recent study showing how the quantification of MS lesions and the registration of all the patients, in this case, over 600 patients registered to the same coordinate space, enables you to, ask questions about the some signatures of MS, of different MS phenotypes, depending on how the lesions are distributed in the spinal cord. So that work from Dominique Eden was then followed up by another work by Anne Kerbrat in 2022. Another application is in amyotrophic lateral sclerosis patients, where here it's not the spinal cord that we wanted to quantify, but the gray matter. And so that's a showcase of the automated gray matter segmentation to compute atrophy in ALS, where we know that the lower motor neurons degenerate, which manifests as a reduction of the surface of the gray matter in the ventral horn of the spinal cord. Another application in stroke patients, where in this case it's diffusion MRI that was used to identify abnormalities in specific tracts, in particular the lateral corticospinal tract and the medial reticulospinal tract. There was a study done in collaboration with Northwestern University researchers. And also, I think one of the first PET MR study done in the spinal cord in patients suffering from chronic pain. And that was, so they used the ligand, PET marker of neuroinflammation, and showing SUV, which is the signal, the standardized uptake values that is used in PET. And the idea of using MRI here is that the MRI combined with SCT analysis provides a localized regions of the SUV in the white, the gray matter, the dorsal root ganglia across different vertebral levels. That was a study done by the team of Marcologia at MGH. And as I mentioned before, more and more studies also looking at fMRI applications in the spinal cord at 3T, but more and more studies at 7T as well. Not specifically related to SCT, but of general interest for the spinal cord community, and all of you present here obviously have an interest on the spinal cord. And so this is a study that proposed a generic protocol to enable researchers, clinicians starting off with the spinal cord imaging, instead of starting from scratch and then choosing, doing some pilot studies to choose the right parameters, TR, TE, type of sequence, matrix size, and so on. We provide some sort of, starting protocol that includes, anatomical scans to look at the spinal cord, to look at the gray matter, diffusion scans, and MT scans. And then this is an example of how this could be used to quantify fractional anisotropy in the spinal cord between C2 and C5 in the white matter. And in the x-axis here, you have all the different sites, and each red dot corresponds to a particular subject. So it really shows the reasonably good inter-subject variation inside, like for a given vendor, but also across vendors, especially when we compare Philips and Siemens. I see that there are some private message for me, so I'll ask you to please post those direct messages to either Mathieu Guépaquet, who is here on the call, or Joshua, because I will not be able to read the direct messages to me while I'm giving the presentation. In particular, this one is about making the window larger, so I can make the window larger. But again, please, if there are specific questions, please redirect them to Mathieu or Joshua, or just post them publicly, and then Mathieu and Joshua will interrupt me if necessary. Thank you. Okay, so some technical details about SCT. It supports finally it supports Windows. I mean, it's been like that for many years, but now we can officially say that Windows is well supported, as well as Mac OS and Linux. And then it uses the command line interface, as you can see here. But we also have some integrating the developments with Facialize, which is a viewer developed by the FSL team. So for this course, we're going to use some conventions, some green labels, if this is something we're going to do together. Okay, so you're on your side, you're going to open a terminal and you're going to type some comments here. If it's red, it means that this is something you can try at home, because we won't have the time to do all the slides together. If it's a yellow thing like that, it means that it's a useful tip. Any questions so far? Okay, let's get into the meat of the subject, installation. So I would like to know how many of you have installed SCT. So I'm gonna create a poll to ask you that. Let's see, more polls. Great. Yes, I'm creating the poll. It's gonna take 2 seconds. Okay, save. Do you see the poll now? Okay, I need to click on launch. Okay, the poll is launched. Okay, let's just wait 30 seconds or 20 seconds. It's a fairly easy question to answer. Okay, awesome. So the vast majority has installed it. For those who have not, what I invite you to do is, install it now. And if you have some issues, you can send direct messages to Mathieu or Joshua, and they will assist you with installation. But briefly, you go to, if you're on macOS, for example, you go to this web page, and then the option 1 is the recommended one. You open a terminal, or like first you go to the latest release, and then you download the script. So Joshua, I think you added a script recently for 10.15, but the one to use for macOS would be this one. Then you download it. It goes on the desktop here. So it says to go to download, but, I download it on my desktop. It's here. So I would just go to my desktop, and then I would launch this thing. Bash installed, bash install, and that's it, right? So I won't do it now. So I'm just quitting, but this is how you would install the SCT. Coming back to the course, you can resize your terminal. I put it on the size of 144. I can make it a bit smaller like this so that we can see the slides. Some very basic Unix commands. So this I'm not going to give you a course on Unix. I'm not an expert in Unix myself, but it's very useful to know the basic minimums of Unix, such as to show hello world. That's the command echo. If you want to see where you are in your folders, you can write pwd. And then if I open a Finder, which is equivalent to an Explorer Windows on Windows, this is my Finder, you see. And if I go to desktop, then this is my desktop. So when it shows users Julian desktop, this is the equivalent of what I see on my visual folder. So there is a bit of a mental adaptation to use the CLI and to remember where we are in the folders. So if you want to go to a particular directory, or if you want to list the directory, you would write ls. So I can see all the things that I'm seeing here, you see. If I want to create a directory, mkdir, nice dir, and then I can go inside it, nice dir. And now I'm inside that directory, and you see that this has been created here as well. And then if I want to go back to the parent directory, I would write cd ... And that's pretty much it. Okay, let's download course material. But before doing that, I need to remove the current ones that I have, because otherwise it's going to be a mess. So I'm going to remove this and this. Oh, okay, it's clean. So I'm going to go to my home folder. So it's a green sticker. So you can all put your hands on your keyboard and then start doing the same thing as I do. I'm going to make my thing a little bit bigger so that you can see it better. Okay, so cd tilde, I'm on my home folder. You can go wherever you want, but it's just so that everybody is at the same location. And then we're gonna write cd download data. Sorry, SCT download data. Okay, so I want to show you something as well. Instead of writing SCT download data, because I can make some typo, that's there is a feature on Unix where if you type the tab key, it will automatize the rest of the command. So if I type SCT down and then I type tab, you see that the rest of the command is automatically appearing. Right? So that's a way to write faster. That's a way to write faster. Another way to do it faster during the course is to also copy the actual command that is there. So one way to copy quickly is to do a triple click. So 1, 2, 3, and you have the entire syntax that is there. Then you do Control C or Command C if you're on a Mac, depending on your OS, and then you do a Command V or Control V, and then you can paste the command here. Then enter. And that's it. So you see that now I have a folder called SCT course data. I can go inside it using CD SCT course data. So I'm going to go CD and then look SCT underscore, and then tab up. Everything is automated because this is the only folder that starts with SCT underscore C. LS, see what's going on in there. We have a README. It's nice, multi-subject, single subject. So we're going to start with a single subject. The CD space single tab, and that's it. Okay. Let's wait a few seconds so everybody is there at the same time as well. All good so far? Any questions? I see. Questions about upgrades. Yeah. Okay. All right. Let's move on. Yeah, so all the comments that we're going to see in this course has also been copied inside a batch file. So it's called batch single subject. We can open it with a text editor, Sublime Text or text edit. I like to use Sublime Text because it creates nice colors. And you see it's basically a collection of a bunch of scripts that we're gonna see on these slides, right? And that same script is also accessible online, by the way, because the whole course was downloaded and it's downloaded from GitHub. So all that is also publicly available on that website. All right. A little bit of background information before we begin. If you want to look at your image and, you know, like every time I have new students coming to the lab, the first thing I say is look at your data. You always have to look at your data. This is the most important thing in medical imaging. It's imaging, these are images, so look at the images. Very, very, very important. Because without looking at the images, it's very easy to just run a processing, get some weird results, and then you know, draw wrong conclusions because there might have been some, you know, some artifacts in the image. It might be an image of the brain, whereas you expect an image of the spinal cord. Some automated segmentation could fail. So everything could happen. So it's very important to look at the images, and there are different software that are out there, free software that you can use. We like to use Facialize in the lab, essentially for historical reasons, but also it's a nice software. ITK Snap is very popular as well. What's nice with ITK Snap is that it works for Windows, which is not the case for Facialize. 3D Slicer is a bit heavier, I would say, a bit more complex. There's a lot of buttons in 3D Slicer, but it's also a nice software that has many analysis features as well. And Horos, so all these software here work for Nifty. If you have DICOM data, you can also use Horos, which is an equivalent of Osirix, but that's essentially for macOS. So if you're using Linux or Windows, you might want to look for alternative software. So by the way, maybe Mathieu, we can add a note to update this slide and add some, you know, DICOM friendly software for Windows and Linux, because it's missing here. Nifty is a file format. It stands for Neuroimaging Informatics Technology Initiative. It's a fairly old format. I think it's been around for, I don't know, 20 years, something like that. I'm not sure. It started at NIH. And yeah, it has, you know, simple header. And it's you know, used by many neuroimaging pipelines. So SCT relies on Nifty, right? So if you have DICOM data, you need to convert them into Nifty before being able to use SCT. Some comment about spaces in file names. Please never use folders or file names with a space like this one, because these spaces will be badly interpreted by some software, depending on the OS, on the operating system you're using. So it can lead to some errors. We've seen that in the past, users, having some issues with their pipeline, and in fact, the issue was just coming from the way the files and the folders were named. So make sure that you don't have any space in your folders, and you can use dash or underscore. All right, so we're ready for the segmentation. Any questions so far? Yes, thanks. 3D Slicer also works with DICOM. So when I said it, I realized, but yeah, good point. Okay. So we now have, so that's actually the first year that we officially replaced SCT PropSeg and SCT DeepSeg SC by a new command, which is now SCT DeepSeg. So that's a big change compared to the previous SCT courses. We call it a contrast agnostic approach. Sandrine is going to describe this in more details. But in brief details, the previous command, PropSeg and SCT DeepSeg SC, were specific to different contrasts. For example, if you wanted to run PropSeg on a T1 weighted image, you would write -C and then T1. And similarly, if you were to run SCT DeepSeg SC on a T1 weighted contrast, you would write something like SCT DeepSeg SC, then -C T1, right? Or T2 if you have a T2 weighted contrast. So that created some issues. I mean, yeah, in terms of the variability of segmentations across contrasts, and it also created some confusion when for contrast like PSIR, for example, how do you call that contrast, right? So I'm gonna stop now and give the mic to Sandrine.
Hi. So not only like having single models per contrast is confusing in selecting the model, but there was also another problem that we found. It was that the segmentations provided morphometric measures that were different across MRI contrast. So for example, we computed the cross-sectional area and we average across the C2, C3 vertebral levels. And then we obtain different cross-sectional area across 6 different contrasts for the same subject. So this is what we see on the top panel. But now with the new model, we have a stable cross-sectional area across the 6 different contrasts. So we can change slide. And so how did we do this? So the idea, instead of having one model per contrast, we tried to unify and create one single segmentation for different contrasts. And to do that, we created an average of the segmentation for each subject. And we got a unique ground truth, and we used that to create our new models so that we have now stable cross-sectional area and morphometrics across the different contrast. And now you don't have to specify also the contrast for the model. It can take any image. So I think now we'll try it out.
Excellent. Thank you, Sandrine. So hands-on. So you all have your terminal. I'm gonna remove this thing. First thing we're gonna do is go to CD data T2. So triple click, copy paste. And let's see what's inside this folder. So ls. Oh, there is only one image. All right. And then we're gonna, I don't want to type all that, so I'm just gonna triple click, copy, paste, and that's it. And so now you can see that SCT DeepSeg is associated with a particular task. In this case, it's the SEG SC contrast agnostic. Okay. There are different tasks in SCT DeepSeg that we're going to talk about later on. The -I stands for input. So the input image is the T2. That's the image that we input in the algorithm. And then you see there is something called -QC tilde QC single sub. That's another flag that tells SCT where you want your QC, your quality control outputs to be located. And that's also something we're gonna see very shortly. So if I scroll down, you see that it tells you a bunch of things. The model is up to date. If it was not up to date, then it would download it automatically. Starts the inference, tells you how long it ran. And then usually when you see a green writing, then it means that this is something useful for you that you can use to visualize your output. You can see that there is one that refers to the QC, right? So I can copy paste this line and then it will open the browser. And then we can look at the QC together. Or you can also look at the image, the input image, which is this one, the T2.ni.gz, with the created segmentation. Okay. Let's look at my Finder window, single subject, data, T2. That's my image. And you see that there is now a new image that was created, T2_seg. I'm going to talk about the JSON file shortly as well. But first, let's look at the results. So I'm going to triple click here and then launch this. So I have Facialize installed. If you don't have Facialize, you can also open the image directly from your Finder. So I'm going to toggle off the segmentation. So that's the the sagittal view of my image, coronal, axial. And then when I toggle on the segmentation, you can see that it does a pretty good job at segmenting the spinal cord, especially in location where in this case this is an asymptomatic very slight compression of the cord, as you can see on the axial view. I'm going to zoom in a little bit. But even though there is a fairly abnormal shape of the cord, the algorithm was nevertheless capable of segmenting it properly. So I talked about a JSON file, this one here. Let's open it to see what's inside. You see that that's also something fairly new compared to the previous versions of SCT. It to be closer to the BIDS standard, which is a standard way to organize the files and to name the files in neuroimaging. It basically creates a file that shows the version of SCT that was used to create this segmentation. It shows also the code URL that was used and the name of the model with a link to the model for segmenting the spinal cord. So that way, if let's say you decide to rerun the same code in 6 months, there's going to be another version of SCT, and then you can have that the segmentation was produced with that particular version of SCT, and you can reproduce it if you want. It reinforces the concept of provenance of your analysis pipeline, which is very important in complex analysis pipelines. Um, any questions so far? Right, there's a question from Marco about running in homogeneity correction before running those pipelines. So this segmentation is a 3D segmentation, not a 2D. And it was trained by using fairly aggressive data augmentation methods that included also, some bias field introduced into during data augmentation. So we think it's fairly robust towards different field bias, as you can see here, where the image intensity is fairly lower compared to there. There are some other extreme cases where we almost don't see the spinal cord, but nevertheless the model works fairly well. So I think this is pretty robust to bias field, but it doesn't prevent you from applying some bias field correction if you feel that this is helping. Question from Ricky Wade. The reason the pipeline wouldn't work on data from children, please. Right. A very good question. I think Sandrine can correct me if I'm wrong. I don't know if Naga is connected, but did we include the data from children as well? No. Okay. But so Ricky, that's a plan to also include different ages into the next version of the models. I feel so again, data augmentation included some scaling. So it's possible that the model is still fairly robust on like smaller spinal cords, but give it a try and let us know. We would be very curious to know how it goes.
Yeah, I know we have someone in the lab working on a template for children, and they are using this model to segment the cord in children MRIs, and it currently works well.
Okay, thank you, Sandrine. All right. So I told you also that there's another output here that refers to the QC, or in other words, quality control. Let's run that command. If you're on Linux, the command would look a bit different as explained in this slide, right? So that's more like OS specific. I'm on Mac, so the command is open, and you see it's pretty fast. It opens a new window on my browser. And then if I click there is only one line because I only run one command, and it shows the spinal cord segmentation with the axial view in a mosaic format. So it really gives me, a 1-second assessment of how well the segmentation works. And if I click on the right arrow, you can toggle on and off the overlay. So you can see now how useful that is. If you have 200 subjects to QC, they would each appear as a single column, as an individual row, and then you can quickly go to the previews of the next subject and then toggle the overlay and then assess if the segmentation works or not. We're gonna go into more details in the QC during the second day of the course. Coming back. That's it. I talked about this already. Right. So, the fact that contrast agnostic is out doesn't mean that the previous algorithms are not, not usable anymore. In some cases, you might find that SCT-PropSeg or SCT-DeepSeg-SC might provide better results, especially because SCT-PropSeg is a very different method. It's essentially a propagation of a 3D mesh, and that is driven by the image intensity. So if you have, for example, ex vivo spine that were extracted, that does not look at all like an in vivo anatomical image that was used to train the deep learning model, then it is possible that PropSeg would work better. So I invite you to, if that doesn't work, try other algorithms, and then feel free also to post on the SCT forum, if you have some difficulties, and we'll be happy to help you with that, because we have some experience with dealing with different types of images. Question from Fozzy. That's Sandrine answered. Okay, very good. And so speaking of DeepSeg, SCT DeepSeg, there are different models out there that you can see here. If you type SCT DeepSeg, enter, it lists all the different models that are available. So the one in green are already installed on my system. The one in red are not. But if you have, for example, mouse, spinal cord, and you want to segment the white and the gray matter, then there's a model for it, right? And we're gonna see some of the models later on in the course tomorrow, in the second day of the course. And we also have this very nice model gallery that is available online that shows all the models with explanations about how the model was created, inputs, outputs, and so on. Any questions so far? So the default model, I would say, there's different definitions of what we call default, but like I would say that my default personally would be the contrast agnostic model. Yeah, it's the new default, which is why we've updated the slides now to reflect this. Okay, so if there is no other questions, we can continue with the second part. Sorry, with the next section, which is about the vertebral labeling. So that slide is pretty old now, but the algorithm that is in use in SCT, currently the default, is to find the C2, C3 disk using machine learning approach based on the Hoff transform and some regularization. And then there is a straightening of the spinal cord that is going on, and finally a sliding window that will identify the different vertebrae. And that sliding window is also weighted by the probability of dislocation based on the size of the spine. And so it's basically like a model that would adjust each time it finds the next vertebrae. Okay, so it's fairly robust in cases where there is missing vertebrae, for example. You can find the details in this publication. And a few words also about what we call labels. Vertebral labels can mean two different things. It can mean the full body labels, which is used for CSA calculation. It can also mean point labels, or we also sometimes call it point-wise labels. And it's a single voxel that indicates where is the middle of the vertebral body inside the spinal cord, right? And that is used usually as a landmark for registration to the template. Okay. And we have some conventions here for the vertebral levels, for the point-wise vertebral levels. 1 for C1, 2 for C2, and so on. Okay. And then we also have equivalent labels for, we call it disks, but you will see that they are not all disks. So C1, C2 would have the value 2, C2, C3 disc would have the value 3, and so on. But we also have the pontomedullary junction, the value 50, and the pontomedullary groove. And also, we've introduced the label 60 because more and more people are doing lumbar processing, and they needed some like the tip, the conus medullaris label in order to use that as a registration method for their lumbar data, right? Because relying on the disc or the vertebrae makes little sense when we look at the lumbar spinal cord. Sounds good. So let's move on to the hands-on. So your participation now. First, we're gonna label the vertebrae. So triple click, copy, and then paste. And then we're gonna follow what the thing is doing. So you see that first it straightens the spinal cord, and it straightens it using, I'm gonna go up here a little bit to explain what I inputted. I inputted the image, okay? But I also inputted the spinal cord segmentation that we got from the previous step. You see? Here we have to specify that this is a T2 weighted contrast, because this is how the algorithm is going to work better. And then we also specify the QC output, which is the same as before. So we should expect that there's going to be an additional line that is going to be created at the end of the process. Okay. Going down, it does the straightening. It creates some temporary files that you can inspect if you want to understand better how SCT works in the background. Resamples 2.5 millimeter isotropic. It finds C2, C3. It detected it. And once it detects it, then it will go basically up to find the disk C2, and then C1, and then it goes down to find the disk C3, C4, C4, C5, and so on. And when there's no more disk to find, it unstraightens, put the label back in the original space, creates all the files, and then you have your green value, your green syntaxes that you can use to QC your results. So I already have the QC window open, so I can simply do a refresh, right? So you can click on refresh here, and then you see that there's an additional line that refers to the SCT label vertebra, and that shows the results of the vertebral level. Again, you can toggle on off to display or not your labeled segmentation, right? And then you can go back. If you type up, down, then you can go between the two different processes. Any questions so far? Okay. Oh, yes, from Lucien. Do we have to have C2, C3 in the field of view? Ah, that's a great question. Right. So since you're asking the question, I'm going to answer it. So the short answer is, if you want to use it as is, then the answer is yes. You need to have C2, C3 in the field of view. If you don't, then label vertebrae. Let's run the command with -h, which means help, and it indicates, all the ways you can use the function. So you see there is a lot of flags that I did not talk about. In this, 2D course, I won't be able to describe all the different parameters. But there is something called, initialize using slice number and disk value. So for example, if you know that that your field of view, is centered at, let's say, T8, T9, and that corresponds to, a Z value of, let's say, 64, then going back to the definition of the disk, T89 is the value 16, you would write something like this. Limit Z, and then that would be, we say that the Z would correspond to 64, and then T89 corresponds to the value 16. So you would write something like this, right? And then it will start the algorithm from there. What you could also do is initialize using another Nifty file that has a single disk label that has been created, manually or by like another software, for example. So there are different ways to deal with that, but yeah, that's longer answer is that depending on how your pipeline is done, what kind of imaging constraints you have, then there are some options. So if you need only lumbar, how can you specify it? Well, so that would be the similar answer. If you have some information of how your lumbar data have been acquired, for example, if the field of view is centered at T12 L1, then this you can use init Z, for example. So that's with the red label. So we won't do it. We won't do it here. I invite you to do it at home, but I can just show you quickly how that's gonna look like. I'm just going to launch that command. So you see that here it opens a manual window, and then you can click on label C2, C3. And let's say that you have lumbar data. You can, if you have lumbar data, then you can run the same commands, but by changing C2, C3 to like T12 L1, for example, and then you would change the 3 for the appropriate label value. For T12 L1, that would be 20. Okay. Does that make sense? Okay. Any questions so far? So there's a question, technical issue with Nabila. So I guess Mathieu or Joshua can maybe check with Nabila what's going on the private channel. Okay, so I'm going to continue now with the shape-based analysis. Oh, that's presented by Sandrine. Okay, I have a little pause here.
Yeah, so we already kind of introduced cross-sectional area, but we didn't have all we needed to compute it. So now that we have the segmentation and the vertebral labeling, we can now compute the cross-sectional area. So here we will average it across the C3 and C4 vertebral level using the flag vert here. And so when using this, it will take the label file and it will average it across those two levels. So Julien, you can run the command. Okay, and we can open. So by default, it creates a CSV file, and we can open it to see what's in there. We also put it in the slide. So we have a lot of information in this. So first we have the STT version, you will have the file name, you will have also the slices for which correspond to the 3, 4 level. Then you will have the CSA, so mean area will be the CSA here. You'll have the standard deviation as well. And also some interesting things. You also have the angle, so AP angle and right-left angle. So if we look back in the slide, we have what it corresponds. So the cross-sectional area is corrected, is used to correct for the angle of the chord. So maybe it's also good to check these angles if maybe there are some different values. Also, when you compute CSA, it's very important to always check your segmentation, because if your segmentation is bad, well, you will have wrong CSA values. So you always want to make sure that your spinal cord segmentation for your region of interest that you want to compute is okay. And also checking the quality of your vertebral labeling to make sure that it's in the right spot. So if we continue, now we average it across C3, C4 vertebral level, but we can also do it per level. So we would add the per level flag here. So when we run this command, Junie can run it. We will get a CSV file now. We will have one line for the vertebral level 3 and one line 4 and one line for the vertebral level 3. So we'll have 2 rows here. And now we have both vertebral levels. Now there's another option. We can also run it per slice. So we will add this specific flag now, and we will get really 1 row per slices that are, and here we also specified the slices. So we'll get from slice 30 to slice 35. So yeah, and we need to see which vertebral level they correspond also. So also notice that for the STD here, there are 0 because the STD is computed when you average across different slices. I see there is a question about if the metrics are computed in the straightened PAM50 space. So they are not. It's really computed in the native space. However, if the spinal cord is at 45 degrees, the slice will not be at that angle, but we really want the cross section of the spinal cord. So that's why we compute the angle along the spinal cord, and we correct for that to account for the angle of the cord, but it is done in the native space. And it's relative to the center line of the spinal cord, if that answers the question. Now we can continue to the next slide. So there are different ways to compute cross-sectional area. You can always check the different flags, but using vertebral levels to compute CSA, it doesn't consider a variation in neck positions, for example, in subject. So to overcome this limitation, we can also compute CSA from a distance from a reference point, like the pontomedullary junction. And then we will compute a distance across the center line to compute the cross-sectional area. So first, we can use SCT detect PMJ to detect the pontomedullary junction automatically. and we can check the QC report to validate its position. So it is good. We can continue now running the SCT process segmentation. So here we will now include our PMJ label file. Then we will compute CSA at 64 millimeters from the pontomedullary junction. And we don't want it on a single slice. So here I want to average across a 30 millimeter extent, for example. And now we can look again at the QC report. And we will see exactly where the CSA was computed, and we can validate the center line. This is where I wanted it. And we will have now our, in the CSV file, we will now have our values for that CSA. And yeah, we can continue. So I think Jan will continue the next part.
Yeah, sure. Hello, everyone. So in addition to the cross-sectional area, the SCT process segmentation function provides also other shape metrics, such as AP diameter, RL diameter, eccentricity, solidity, and orientation. And all these shape metrics might be particularly relevant, for example, when studying spinal cord compression in conditions such as degenerative cervical myelopathy. The images on the left, they show an example what such a compression can look like. So we have a sagittal and axial image of a subject with spinal cord compression. So the compression is highlighted by the red arrows. And if we now take a look on the charts for individual shape metrics, so we have the level of compression highlighted by these red dashed lines, and you can see how the shape metrics are influenced by the compression. So for example, we can see the decrease in cross-sectional area and in the AP diameter. And all these other shape metrics are saved together with the cross-sectional area into the output CSV file. Okay, I believe we can move on to the next slide. Yeah. The SCT process segmentation function also allows you to normalize the shape metrics to the PAM50 spinal cord template. Just very briefly, PAM50 stands for the spinal cord template. This template will be covered in details in the next section. So for now, the PAM50 template is just an image created by combining images from multiple subjects, and we can use this image as a reference space when we need to analyze multiple subjects. So the figure on this slide illustrates how the process works. So first, the function automatically computes the shape metrics in the native space, and then the shape metrics are interpolated per vertebral levels to the template space. So let's say that you have 20, 30, 40 images and you would like to compare all of them together. So you can normalize or bring all of the images from your individual subjects to this template space. This normalization is done using normalized PAM50 flag. The normalized metrics are saved into the CSV file, as is illustrated at the bottom of the slide. And once you run the command, you will notice that the slice numbering will be different in this case because the metrics will be normalized to the template space. So the slices here in the inferior superior direction will correspond to slices in the template space. And if we move on to the next slide, here we have an illustration what we can do once we normalize our metrics to the template space. So you can compare the normalized shape metrics with the normative database. So this figure is showing cross-sectional area. So on the vertical axis, we have cross-sectional area, and on the horizontal axis, we have individual vertebral levels. so C1, C2, C3, and so on. And in blue, we have mean and standard deviation values of cross-sectional area computed across a normative database across 203 healthy subjects. And you can see how the cross-sectional area is changing across levels, which is expected because the spinal cord anatomy is not the same. And since we normalized our single subject metrics to this PAM50 template, we can now plot our single subject data together with the normative database. And we can kind of see what the cross-sectional area looks like together with the normative database. Yeah, I believe we can move on to the next slide. Here we have another possible application of the normalization to the template space. So here we have a subject with spinal cord compression at the level of disk C5, C6. And this figure is showing AP diameter. So basically the diameter of the spinal cord in the anterior posterior direction. And again, here we have the normative database. So in blue, we have AP diameter computed across 203 healthy subjects. And in red, we have AP diameter for this particular subject. And you can see how the AP diameter decreases at the level of compression, so at the disk C5, C6. So this basically means that the spinal cord is compressed, squeezed by the compression. And yeah, this is preoperative scan. And if this subject was scanned also after the surgery, you could kind of plot both measures into the same figure, because now the shape metrics are normalized to the PAM50 anatomical dimensions. So this is one possible application. This normalization can be used to compare measures from many different runs into a single visualization. Okay, moving on to the last slide of this section regarding shape metrics. Here we have another function which is related to shape metrics. This function is called SCT compute compression, and it allows you to compute metrics to quantify spinal cord compression. These metrics are based on so-called MSCC. MSCC stands for Maximum Spinal Cord Compression Index, and it uses this equation, this formula. And basically, the function computes the metrics, in this case, AP diameter at the level of compression, and it normalizes it using AP diameter above and below the compression. So it is kind of self-normalization. And additionally, we also implemented an option to normalize using a database of healthy controls. So you can also compute so-called MSCC norm, as is illustrated on the right. So all these single subject, the shape metrics, are normalized relative to the database. And this aims to reduce intra and inter-subject variability. And importantly, you can also filter this database according to sex and age. So let's say that your subject is female. So you can select only females from this normative database, and then your shape metrics will be normalized according to females. And yeah, one important thing here we illustrate only AP diameter, as was originally proposed in this method for MSCC, but now you can normalize any metrics, any shape metrics provided by SCT. So you can use this MSCC formula also for cross-sectional area, RL diameter, or any other metrics provided by SCT. Yeah, if you need more details, please visit this tutorial. Yeah, and that's it.
Great. Thank you, Jan. Thank you, Sandrine. Moving on to pretty like a core section of the course, which is a registration to the template. So a little bit of theory. A template, it's a commonly used concept in brain and neuroimaging studies. And it essentially refers to an anatomical average of a given population. So the PAM50 template is a template specific to the spinal cord. that is represented as a straightened spinal cord averaged across 50 individuals, which is what the 50 comes from. And it stems from a collaboration between Polytechnique Montreal, our institution, Aix-Marseille University, and collaborators at the MNI, the same who created the famous brain MNI templates. And what's nice is that the PAM50 template is actually located within the same coordinate space, the absolute coordinate space of the actual ICBM 152 MNI template, right? So if you interested in doing brain spine studies, then you can use the PAM50 and do your separate analysis of the brain and end up within the same coordinate space. The PAM50 template comes with what I call objects, but essentially these are labels of the vertebral levels of the white matter probabilistic location, gray matter as well, cerebral spinal fluids, the different white matter tracts, as well as gray matter parcellation. There are 6 different parcellation of the gray matter also, the ventral, median, and then the dorsal parts of the gray matter, which can be used especially for fMRI studies. We also have a spine template, which does not appear here. Maybe we can add it in the next course, Mathieu or Joshua, a note for next year. So how does the registration to the pipeline work? This is a brief overview. It's like you have a subject. This is the same subject that I showed you before on Facialize. We're going to segment it. We're going to identify vertebral levels. These will be used to then, after straightening the cord, find the affine registration to match the vertebral levels between the subject space and the PAM50 space. And after that affine transformation, there is a non-rigid transformation to align, the final details of the spinal cord of that subject onto the PAM50. Then all the warping fields and transformations are concatenated into a single file. And then we can use that to bring all the objects, the labels into the subject space, and the other way around. So we're gonna go through this process together. First, we start with the segmentation. We did it already. Vertebral labeling, we also did it. Straightening. The straightening is an algorithm, which method has been explained in that paper from Delenier et al. Can show you a fancy video. Oh, actually, the video is accessible here. Is it? I think I need to connect. Yes. Yeah. So it shows that the algorithm is using B-spline plate interpolation to really like bend the spinal cord into a more straightened representation, right? So this is how the PAM50 template was generated. We did it for 50 subjects, and then we did the affine registration and fine registration, and then average. And then if you want to register a single subject to the templates, then we follow the same procedure, right? Okay, and then the cross-sectional area could be measured along specific vertebral levels. We can also compute we can also infer on the cross-sectional area of the gray and of the white matter. And we can also use the labels of, coming from the white matter tracts or the gray matter parcellation to compute quantitative MRI metrics such as magnetization transfer ratio or diffusion-weighted MRI metrics coming from DTI model, like the radial diffusivity, fractional anisotropy, mean diffusivity, in specific white matter tracts. So that's, you know, one of the typical applications of registration to a template. All right, so after that little theory, let's put our hands on the keyboard, reopen the terminal, and then run some of those commands. So we are still on the T2 folder. And what we want to do now is create labels at C3 and T2 mid-vertebral levels, right? And we are going to create them using the labeled segmentation that we already created by running the SCT label vertebra command, right? The SCT label vertebra command is the one that generated this file here, right? So we're gonna extract the center of mass of this level, C3, and T2. We're gonna create 2 points here. Okay. So let's run it. So that's a pretty fast command. And then we can look at the results. Actually, do I have a QC? I don't have a, or maybe something. Oh yeah, we can generate a QC report. But I thought that we introduced the QC report in the recent version. Let me just check something. Oh yeah, we did. Okay, so maybe Mathieu or Joshua, note for next year, we can actually run the QC directly from that command, and we don't need to generate a QC report from there. So that's an update on the slide that we can make for next year. But anyway, I'm going to run that command so that we can generate a QC report based on the labels that I just created. Okay, we can open the QC report or refresh the existing one. Refresh, and you see now that there are 2 point-wise labels, number 3 for C3 levels and number 9 for T2 vertebral levels. Okay. And this file is called, this file is actually called, where is it? I called it here. It's called T2 labels vert, right? So that's the one. So if you open this one on Facialize, you will also see those 2 points. Next slide is just to show that there's an alternative method. If you don't have any vertebral labels, then you can also create those labels manually. Similarly to what I showed you before, you can run the SCT label utils command with the create viewer option, and a window will pop up, and then you can manually click on the intervertebral discs of interest. Okay, so I'm not going to run that here, but this is something you can try at home. And finally, we're going to run the key command here, which is register to template. We're gonna input our image, the T2-weighted image. We're gonna input our spinal cord segmentation, T2 seg, and we're gonna input the labels at C3 and T2. And with these 3 inputs, it will do all the job. So let's try it. Triple click, copy and paste. So that's a relatively slow process because there's a lot of things going on here. It will reorient the data to right posterior inferior orientation. It will resample everything to 1 millimeter isotropic, straightening of the spinal cord, which is usually a relatively long process. It runs a step 0. The step 0 is essentially the step that I showed you before, which is going to go back. Step 0 is this one, this affine transformation. Okay. And then after step 0, there are like step 1. It does a center of mass registration based on the image and the segmentation. So it will basically, after the cord is straightened, it will take the center of mass of each point of the segmentation, and it will try to align it with the PAM50 template. And then there's a second step here, which uses the segmentation of the cord. Okay. So let's say if you have a subject with a compressed spinal cord or with an atrophy, then you need to account for it in your registration. And the segmentation will appear, will reflect this abnormal morphometry, and it will use a regularized B-spline scene transformation, which is coming from ANS software, to register the segmentation of your subject into the segmentation of the PAM50. So that process is the longest one. And you see that after that, it estimates the transformation, and then it will concatenate all the transformation that were run for step 0, step 1, and step 2. It will do it for the forward transformation. So if you want to go from your subject to the template, but it will also do the inverse one if you want to go from the template to your subject. So you end up with 2 different warping fields. And if I open the Finder window, you're going to see them. Oops. That's my Finder window. You see, there is a warp anat to template. It's a warping field that brings you to the anatomical image to the template, and you have the inverse one. And then you have your, traditional QC report command, and then you have also the Fesselize command to look at your native space, so the template registered into your native space, or your subject registered into the PAM50 space, right? So we can look at all of them. First, let's look at the QC report. Refresh, new line, and then it shows the axial view because this is the most convenient one. And then we can toggle between the image. So that's the image of your T2-weighted scan in the space of the T2-weighted scan. And that's the registered PAM50 template. And we can toggle between the two, right? So again, it is a quick way to assess whether the registration is satisfactory or not. And you can do the same also by inspecting more the image by opening your viewer. So that shows the registered PAM50 T2-weighted image. I'm just going to increase the dynamic a bit more. And then if I toggle it off on Mac, I can use Command F. On Linux, it's Control F to toggle it on off. I'm going to adjust the dynamic a little bit. That's it. Like this. Okay. So you see, yeah. So that's pretty much it for the registration to template. Let's see, where were we? We were there. Okay. Any questions so far? This is a pretty important piece of the course. So if you have any question, feel free to ask, please. Feel free to also raise your hand and then talk directly into the mic. Ajay has one. I may have missed it, but you need to do any sort of Jacobian modulation when dealing with spine compression, right? So the Jacobian modulation would be to adjust the intensity of the image in case of high compression or stretching of the deformation field, and this is something that is taken care of by Hans applied transform algorithm. So there is indeed an account for the modulation of the signal intensity when applying a transformation. Yeah, that's a very good question. So let's talk a little bit about all the little parameters that you can play with SCT register to template. The first point is about a flag called the ref. If I open my terminal and I type SCT register to template, H, enter. There is a couple of parameters. One of them is ref. And you can choose between two values. One is template and the other one is subject. Okay. So by default, oh, by the way, the default is empty here. Oh, what is that? Oh yeah, no, sorry. The default is here. Default is template. Okay. So by default, it will use the template space as the destination space for the registration. If you were to choose subject, then the destination space will be the subject. And the main difference here is that if you select subject, then there is no straightening happening. Okay. So then you would ask, well, why should I use ref subject then? For example, if you have an image that is an anatomical image that is acquired with highly anisotropic resolution, for example, 0.7, 0.7, and 5 millimeter, like an axial image, then that will create a lot of through plane interpolation errors if you were to straighten that anatomical image. And in such case, it might be better to avoid doing a straightening and instead registering your PAM50 onto your unstraightened subject space. A couple of things to consider here is that you don't want ideally a large curvature because that registration to the subject space will be there are different algorithms, but one of the algorithms is to do a center of mass alignment. So let's say if you have your PAM50 that is straight and you want to register it to a slightly curved spinal cord, then it will take each line and then it will translate it to the curved space. Okay. That is also prone to some errors in terms of partial volume and in terms of the conformity of the spinal cord. So there's a lot of small details that are at stake when you run the non-straightened version of the registration to templates. Some of those considerations are fairly complex. So if you're starting with SCT and image analysis, you might not be able to understand all of them perfectly well, and in which case we encourage you to open some discussions on the forum. If you're not sure what reference you should use for your images, open an issue and then we can discuss that all together. Another thing to consider also is, you remember when the processing was running, I told you, oh, this is this is step 0 and this is step 1 and then this is step 2, and then on step 2 you have it's using the segmentation, it's using this algorithm, it's using this metric, and so on. because these are the default parameters. But you can also bypass the default parameters and choose your own preferred registration parameters. So now it gets also a little complex. You need to get a fairly good understanding of all the different algorithms that exist, how they work, what they are good at and bad at. But very quickly, the ones by default would be the center mass rot, which is based on, which is basically doing a translation and the rotation of the spinal cord based on the image and the segmentation. And the B-spline scene, which you remember was listed here at step 2. And it's basically doing a slice-wise adjustment of the cord shape based on the segmentation, right? If you tell me that you don't have a reliable segmentation and you prefer to rely on the image, then that's fine. What you would do here is you would use the, the flag param. I mean, we can do it together. Basically, right here, you would add param. You would specify, step equal 1. The type would be the segmentation, like say imseg, and then you would use algo equals center mass rot. So we're not touching that, but the only thing you're touching is the step 2, where instead of using the segmentation here, you would use the image. So I'm typing in, right? And let's say you don't want to use the B-spline scene, you want to use something a bit more aggressive in terms of degrees of freedom. So you can use algo equals scene. And if you're using the image, then obviously you don't want to use the mean squares, right? Because you need a cost, like a metric to assess the similarity of registration that is used by the cost function to be adapted to your image. So mean squares is not adapted to an image, but what you want to use instead would be the cross correlation or the mutual information and so on, right? And then you can also specify number of iterations, smoothness, and so on. Okay. So I invite you to try many of these parameters, and after that you can look at your QC, and then you can try many different parameters, and then you compare through your QC, which one looks better. And the nice thing with the QC is that it also, you have a display of all the commands that you run. So if you forgot which one looks nice, then it's basically the syntax is written here. Any questions, comments so far? Okay, so let's move on. Again, yeah, some of the things that I talked about already, right? About the type. Yeah. Actually, it's a green sticker, so that's something we do together. Good. So let's copy this thing here, paste it. And while it's running, I'm going to explain to you what is different in this one. So what we did here is step 1. We used, you see, we replaced step 1 by type equals seg and also algo equals rigid. Whereas by default, it was this thing, center mass rot and imseg. So rigid, it's very simple. It only does X translation and Y translation. That's it. So there is no rotation. And step 2, we are using the segmentation. We are using the metric CC. And we are using slice-wise 1, whereas by default, slice-wise is 0, which means that it's using the information from the adjacent slices to estimate the deformation. Whereas if you use slice-wise 1, it will register each slice independently from the other slices. Okay. And we are adding a third step as well. You can do that. You can add 25 steps if you want. And in this one, we're using the image, cross-correlation matrix, the scene, which, as I said before, is more aggressive. Slice Y is 1, iter 2. Let's see where we're at. Still registering. Step 3, that's the, that's the longest one, which is why I also put only 2 iterations for the purpose of this course. Yeah, so Marco had a good question. That was well answered by Sandrine, indeed. Question from Barbara: if we want to register 2 anatomical images across visits or across subjects. Right. In this case, I think you could use a SCT register multimodal, which is something we're gonna see in the next slides. Yeah. Okay, so it's concatenating. So let's just wait a few more seconds, and then we can look at our QC. All right, QC is generated. Let's look at it, refresh, and then you see I have a second line now with all the commands that I wrote. And what's interesting at this point is that we can compare the 2 different sets of parameters. And you can see there's a huge difference between the quality of the registration, right? So it really shows that try to go beyond the default parameters that are proposed and explore a little bit depending on your images. And one of the things that is interesting to notice is that on the second one, you see that there is a rotation of the spinal cord that is not accounted for. If we look at our subject, the cord is rotated because of this very subtle, non-symptomatic compression of the cord. And that is not accounted for properly by step 1, because you remember what we wrote on step 1 is that we use the algo rigid instead of using the center mass route, right? So it's very important to, if you want to account for the rotation, then you might want to consider another one than rigid. But in some cases, the rotation is irrelevant or would lead to bad rotations if there is not enough information, or if you're in the lumbar region where the cord is more round. In those cases, maybe you want to reconsider using center mass route. So again, feel free to spend a few hours playing with those parameters and gain some intuitions about what those parameters do. Another thing as well is that what I wrote here on the command is I used -L. -L refers to the vertebral labels. If you don't have vertebral labels, but you prefer to use disc labels, then you can also use L-disc instead. And in this case, you would do the registration, but only using the disc labels as opposed to the vertebral labels. That's perfectly possible. An example, the reason we introduced that is because a couple of years ago, we had a group of researchers who acquired very, very small field of view. The field of view only covered a few slices centered at C3, C4. So they did not have a vertebral level. So we created that feature so that it's also possible now to register to the template using a disc label. Sounds good? An example of, adjusting the parameters in a case of, highly compressed spinal cord is you want to use a specific algorithm called column-wise that will really make a good job in, adjusting the shape of the spinal cord in case of high compression. Again, this is just another example of how by playing with the parameters you can achieve, better results, depending on your images and the pathologies that you're working with. Okay. Before lunch, before the break, I think that would be a good stop to talk about this. What can we do with our registration results? So let's first explore what the PAM50 has. So we're gonna go to our SCT folder, our SCT. So I'm going to open a new window. My SCT is installed here. It's under code, Spinal Cord Toolbox here, data, and then PAM50. And you see there is a couple of folders here. One is the atlas. It's the white matter atlas, and it has all the different tracts of the white matter. You see, they are all described in this info label file. You see, there is also a template folder that has all the basic images that I showed you before, the PAM50 T2 template, as well as the PAM50 T1 template. There is also a PAM50 T2 space. Sorry, T2 star, not T2 space. The white matter mask, the gray matter mask, spinal cord segmentation, the vertebral and the spinal levels. We have also the spine. So all these are described in the info label file. And what we want to do at this point is to bring all these labels into the space of our native image. And to do that, we're going to use a command called SCT warp template. So I'm going to triple click, copy, and paste. And the input is the -D. D stands for destination. The destination will be the T2 image of your subject. Okay. An important input is the warping field, which brings the image from the template to the anatomical space, which in this case is your T2 weighted image. The flag -A0 is something that I will tell you a little bit later. And then we have our traditional QC flag. It's running, generating the QC report, and then generating also a facialize command. So let's look first at our QC report, and then we can use our facialize command. Refresh. We have a new line here that shows the overlay of the white matter from the PAM50 template that is now registered to our native T2 space, right? So we decided to show the white matter because it's a nice way to inspect if the border of the segmentation corresponds well to the border of the spinal cord on the subject. And then we can also run that command. It will open up a bunch of images and labels that helps with a more thorough inspection of the quality of the registration. what you have in, I'm going to remove that one, we don't need it. We can zoom in a little bit. And then we can remove that. Okay. So I just removed the, so I'm going to remove all the labels. This is showing the PAM50 T2. And then that's showing the white matter, sorry, the gray matter. We're going to, let's see, very well. I think I have a bug in my, I have a bug in my face lines. It already happened in the past. Right. I'm not able to change the display, unfortunately. Yep, that's a bug. Anyway, I cannot change the intensity, but you can see the blue that corresponds to the white matter and the red corresponds to the gray matter. Okay. So a very important point that I want to make here is that the gray matter you're seeing here is not the gray matter of the subject. It's the gray matter of the PAM50 that is registered into your subject space. It's possible that your subject has, a slightly smaller gray matter or with a slightly different shape, and that would not be accounted for by the analysis that you just did. So it's very important to keep that in mind, because if, let's say, you want to, compute gray matter cross-sectional area in this subject, then you should not use this registered gray matter, because it just corresponds to the scaled probabilistic gray matter represented by the PAM50, scaled down to the size of the cord of that particular subject. But it is not specific to the gray matter shape and size of that particular subject. I hope this is clear. A note about subject-centric analysis. The PAM50 template here was warped to the subject's coordinate space. Okay. As I mentioned, you can also warp the subject files to the template space. In this case, you will use the other warping field, ANAT to template, as opposed to template to ANAT. And it depends the kind of analysis you want to do. For example, if you want to run a group level study of fMRI results or diffusion weighted results, you want to group them into and you want to visualize them. You want to see the average FA map, for example, across 200 subjects. Then in this case, you should do the template-centric analysis. So what we did here. But if you don't want to resample your quantitative MRI metrics into the PAM50 template because you're afraid of introducing interpolation errors, then what you can do is bring your template objects to the subject space, which is what we did here. So yeah, I confused. What we did here is this. It's not that, right? What we just did is we looked at the PAM50 objects into the subject space, right? And that's good if you want to extract metrics on an individual subjects. You extract those metrics, and then you can take those metrics and average them together or build some statistics across a population, right? But if you want to do some mapping, then you would use the template-centric approach. And some technical comments on the warping field that we used. Those warping fields are compatible with ITK, which is a you know, a software ecosystem that is notably used by ants, by 3D Slicer, and it has 5 dimensions. We can look at it if you want. All right, if I do SCT image, SCT image can get you the header, right? So SCT image dash I and then the warping field, template to anat dash header. It's essentially the same as SCT, FSL HD. It shows the header of the file. So if you look at the dimension of those warping field, you have X, Y, Z, and then you have another dimension that corresponds to the which one is this one actually? It's supposed to be 5D, yeah. So anyway, that's the vector field that you have a vector coordinate, so XYZ for each of the XYZ coordinates. So this is why you have a 3 here. Not sure what is this thing here. Not sure about that. Yeah. Anyway, if you want to decompose your vector field into X, Y, and Z vectors to make them compatible with FSL or SPM. You know, if you run like second level analysis on SPM that requires the transformations, then you can decompose those ITK specific warping fields into 4D, you know, FSL or SPM compatible warping fields. And for that, you would use this command. Again, if you need help with that, look on the forum. We have a couple of discussions on that. You know, you can search for, you know, warping fields in the forum. And if there is no answer to your question, then you can ask that. And we will leave that for after the lunch. So I realized that we are actually pretty good with time. And I'm thinking that maybe today we will probably have time to do other things than what's listed here. So the agenda is a bit changed. You know, we might probably touch base on the gray matter segmentation and atlas-based segmentation today. And I think we can also have a slightly longer lunch. Let's aim maybe for 45 minutes. Okay. So we see each other at 12:45 EST, Eastern time zone. So I'll leave the Zoom open. I'm going to turn off my camera and my mic, but you know, see you in 45 minutes. Hey, recording is on. So I hope you had a good break. I guess, I mean, we call it lunch for us in Montreal, but you know, depending on where you are, you might have had late dinner or something. Maybe before we continue, is there any question? Feel free to take the mic also, makes it more interactive. Could be a very general question as well. So one question from Chris. What could cause the PAM50 registration to run for more than 12 hours. Well, I would say a PC from 1984 could be one of the cause. If you have also an image with so you know if you tweak the steps of registration, you put a lot of iterations, for example, that could definitely delay the you know, like make the processing much longer. The size of the image has a lot to do with it as well. The size in terms of number of voxels. Yeah. Any other questions? Maybe I'm scrolling through the previous questions to see if there are some that were not addressed. I see all of the questions were addressed already. Okay. All right. Yes, a mention about saving the chat. That's a good point. Maybe adding a note, Mathieu, Joshua, adding a note so that I don't forget to do it. Thank you. All right, let's let's continue our course. So, what we've done so far is we've segmented the spinal cord, we've labeled the vertebrae, and then we registered our subject to the PAM50 template. And then we got all the labels from the PAM50, and then we warped them back into our subject's native space. But typically, when you run some spinal cord studies, you also have other contrasts, such as DTI, MT, you know, fMRIs, and so on. And so the key here is that you want to register the PAM50 template to those additional scans. So we're going to see now how you do it. Let's say that, you know, you acquire a magnetization transfer image. So we're going to process it. So put your hands on your keyboard, and then we're going to do it together. We're going to go back to the parent folder. S, and then these are all my folders, and then we're going to enter that directory, the MT directory, cd MT. And then we're going to segment the spinal cord there. So let's just do an ls to see what's in there. You see there are two scans, MT0 and MT1. You can look at them quickly. Let's just the one, MT0, MT1. So that's my MT0. You see these are like 5 millimeter thick slice, axial orientation, 0.9 millimeter in plane. And that's the MT0 and MT1, which is the same scan with the offset, with the off frequency pulse that is played out before the imaging pulse. Right, so it's an off resonance pulse that triggers a magnetization transfer effect. So we're going to run the deepseg command again, but this time on the MT1 contrast. The reason we pick the MT1 is you're going to see why. If you compare the MT1 and the MT0, you see that, actually in this case, it's pretty fine. But I find in general that the MT1 shows a better contrast between the cord and the CSF. I mean, in this case, the MT0 is pretty good, but if you look at the slices above, the contrast between the cord and the CSF is not that great, whereas it's slightly better with the MT1, right? Okay, so it's running the inference, and then it's done. So we can look at our QC report. refresh, and that's it. So now we have much less slices compared to our T2, which is normal, right? Because we have like 5 millimeter thickness in this case, and then we can toggle on off. You can see the nice gray matter. And then yeah, segmentation looks pretty good. So now we go to the next slide, and then we're gonna create a mask around the spinal cord, and you're gonna see why we use this mask. So we use the function create mask, and the input is gonna be the MT1 image, because we want to create the mask on that image. Dash P is the method to create the mask. So we're going to use the center line of the segmentation of the MT1 image. And we want our mask to be 35 millimeter diameter and to be of cylindrical shape. And we're going to call it mask underscore MT1. So now we can look at the output of our mask. We're opening Facialize. And this is my mask. I can put it in yellow. So you see that it follows the segmentation of the cord. We can actually bring the cord there. So I'm going to add the segmentation of the MT1. Put it in another color. So in red is the segmentation of my image, of my spinal cord. And in yellow is the mask that I did around it, right? So the mask is really centered on the spinal cord. So we have, oh, you're having a problem here. Interesting. Expected one argument. Okay, so I guess Joshua or Mathieu can follow up with you on that. Okay, so now that we have our mask, we're gonna register our template to the MT1. And in this case, we're not going to use SCT register to template because we've already done that. But we're gonna use register multimodal, okay? And I'm going to explain to you a little bit what's going on in there. So the input will be the PAM50 T2 image, okay? That's the input of register multimodal. We also have another input, which is the segmentation input, which is why it's called iSeg. And it's gonna be the PAM50 cord segmentation, okay? The destination is the MT1, and the destination segmentation is the segmentation of the MT1, okay? So far, so good. We can also input a mask, -M, and then the mask that we've created. And the reason we use that mask is because we're gonna enforce the algorithm, the registration algorithm, to only register what's inside the mask, because we don't care about the muscle, the neck fat and all those things. We don't care about that. All we care about is the spinal cord. So inputting that mask will constrain the algorithm to find the best registration inside the mask and ignore what's outside of the mask, okay? Another important aspect is the initialization of the warping field. Because you remember, we've already registered the T2 anatomical image to the template. And we're going to use that information, we're going to use that warping field to initialize the transformation. And if we don't do that, then it's completely going to fail, right? Because the image, the MT image, is not registered to the PAM50. So we're going to use that pre-registration that we calculated before, and we are just going to refine the registration of the PAM50 based on a specific feature of the MT, and namely it's segmentation. Okay. You notice now that we are using center mass instead of center mass rot because the rotation of the cord has already been accounted for when we did the T2 to PAM50 registration, right? And another feature we're going to use is the segmentation. Again, slice-wise 1, details 3, and that's it. Okay. So what we assume here, we are assuming something very important. We are assuming that this MT image was acquired in the subject during the same session, right? So usually an MRI session is what, 45 minutes, like 30 to an hour, 30 minutes to an hour. The subject lies down on the scanner, they try not to move, but obviously they move a little bit, right? If they were not moving at all, if they were dead, then there was no need to add those special registration steps because the MT would be perfectly aligned with the T2. But it's not the case. Subjects moved a little bit, they tilt their heads, and so on. So that's why we want to add those extra registration steps specific to the MT. Does that make sense? So we're gonna triple click, copy paste, and then it's running it. First step, second step. So you see it's a bit faster now, and the reason it's faster is because we have much less number of slices. And also we have a mask. Concatenating, and now it's nice because it concatenates the transformation of the initial warping field. So PAM50 to T2, and then T2 to MT. And it creates a warping field and it's called warp template to MT, right? Yeah, let's look at our QC report. Refresh. That's our MT image, and this is our PAM50 template registered to the MT now. I'm just gonna lower my hand because Zoom now has some AI method to automatically find if someone raised the hand, and for some reason it's just keeps it that way. So hold on. How do I lower my hand? I don't know how to lower my hand. That's a bit stupid, man. Ah, yes, lower hand. Good. Okay. So you see that the registration is quite satisfactory. What I like to do is, because visually, when there is no clear cerebrospinal fluid around the cord, then it's difficult to appreciate the quality of registration. So what I like to do is just put my cursor in some key aspect of the cord, like here, for example, upper right corner, and then I would just alternate just to make sure that the border are correctly aligned with the actual segmentation. So what you can see here, for example, is that the registered PAM50 is slightly bigger than the actual spinal cord of that subject. It's very subtle, maybe 1 pixel or 2, but it's just to show that you can do better, by tweaking the parameters, using more iterations, and so on. All right. All right. So there's a question from Nabila. Yeah, in case of a lesion, I would also suggest to create a mask, but irrespective of the presence of a lesion or not, it's always helpful to create a mask. In case of lesion, it's possible that the segmentation of the spinal cord would be less robust. So you want to double check that the segmentation is correct if there is lesions in the spinal cord. All right, so this slide is about the question of, can you register to the PAM50 template if you don't have an anatomical image, right? Well, in this case, we started off with a T2, and then we used register multimodal. But you could also directly register your empty image or whatever image you have on the template, exactly the same way as we did before with the T2. So you have to create labels. In this case, for example, we are using only one label, in the middle of the field of view, corresponding to C3, C4 disk, and then running the command SCT register to template using the disk label. And then here we use reference subject as opposed to reference template. So the PAM50 is registered to the subject and not the other way around. And some, custom parameters. Right? So I'm not going to run that. This is a red sticker, but that's something you can play with at home if you wish. All right. So at this point, we got a warping field, and then we can use that warping field to bring the PAM50 templates and labels onto the space of the T1. So we're of the MT, sorry. So we're gonna copy this, paste, and then it's running, it's warping the template for every object. And then I guess it takes a little bit of time, so maybe I can take some questions. Yeah, I'm just reading that the issue that you had, Aurore, is related to like a multi-line command. That's interesting. Okay. Something we'll look into. So Marco has a question. Is there a command to align MT and T2 directly? Yeah, exactly. So let's look at it together. I'm going to open a new terminal command here, like a new terminal window. And so that's what we wrote here, right? This guy there. What I can do, instead of inputting the PAM50, then we can simply input the T2, right? So I would specify T2, and IGD. And then the segmentation would be the segmentation of the T2. 2_seg and I.GD. Okay, the destination is the same. That we can keep the same. So we all keep the rest the same. I just don't want to mess up with my current processing. So I'm going to also specify another output folder for all this, right? How do you specify an output folder? I forgot. So I'm just going to type -H, and then it's going to tell me how to output a specific folder here, O folder, right? So I'm going to do this. O folder, test reg T2. Oh, I think I messed something up. Yes. I'm just gonna stop here. I made a mistake. Can someone notice the mistake that I made? That's a good quiz. And of course quiz. Yeah, I still have the mask. Just going back to deep set. Oh, yeah. So let's address your question later, Gabriela. Any idea, someone? What did I do wrong here? Warping field. Haha, we're getting close. Can you elaborate? Feel free to speak on the mic, huh? There seems to be a rotation that is missing. Not really. You are very close, Marco. I think you should continue on your intuition. Correlation on empty on and empty off. I don't know. That's something else that we're gonna see later. Because here, what we're trying to do again is, to take this T2 and register it to the empty one. If not using the template, then the specific warping fields to be concatenated must be specified. That is, so it has to do with warping field. I'm not exactly sure. Yeah. I'm not sure this is the right answer to my question, just putting it here. It's not really a question of concatenating, but can you like Spandana and then Marco, you're pretty close to the right answer, but you're kind of circulating around. If you can tell me what's wrong in my syntax here. There's something that I should remove.
Julian, I'm going to unmute myself.
Yes, Marco.
I think you're going from the anatomical to the template, but
Yeah.
you remove that one. You need to work.
Yeah, exactly. This guy here, that's Sahar also mentioned in the chat. This thing here, we put it because we used the previously computed registration to go from the template to the anat. But we don't care about that. We don't care about that here because all we want to do is register the T2 to the MT, which is in the in the same like absolute space coordinate system because the subject was scanned within the same session. The T2 and the MT1 were acquired back to back. So, with the subject not moving too much. So the absolute coordinate system is the same, right? The matricial, the voxel coordinate system is not the same because these images have different resolutions, but the absolute one is correct. And maybe that's a good segue for me to explain what I mean by absolute coordinate system. If you look at an image header, so SCT image MT1 and then header, you see that there is something called Q form and S form. These Q form and S form are matrices that includes a rotation matrix and a translation matrix that brings basically your like 3D volume into the isocenter of the scanner. So it's really a way to register to know where your field of view was acquired with respect to the coordinate system of the scanner, which is what we call a physical coordinate system or an absolute coordinate system, right? So when you acquire your subject, back to back, subject in the same position, then thanks to the Q-form, you can be sure that, those two images will be, pretty much aligned. And you don't need a, just going here, just going back to my initial, sorry, it's a bit of a mess. You don't need that initial warping field, right? So I'm going to rerun that command without it. All right. Does that make sense? Okay. Refresh. I have another one here, and then this one is showing our T2. So that's not the PAM50, but that's actually the T2 of our subject, right? You see how well they are registered? Well, it's no surprise because it's the exact same subject, right? So the registration is pretty good. Whereas the one that we did before is the PAM50 T2 on the MT1, right? So you see the image is different, right? Here we're looking at the PAM50. Here we're looking at the T2 of the subject. They look the same because they are both T2 images, but one is an average and then one is the actual image. Sounds good? So that was a good question. All right. Okay, so warp is something that we did, and then we can run Facialize to look at our results of the warping. So I don't have the weird bug anymore on Facialize, so I can adjust the opacity, make it brighter. I'll remove this one. And then you can see now that we have can remove the gray matter. And then we can toggle on and off between the white matter, which is coming from the PAM50, and the empty image. Now we are ready to do some quantitative analysis and use this white matter to extract, MTR, for example, or other useful metrics. But before doing so, we need to compute those MTR. And in order to compute the MTR, then we need to register the MT0 and the MT1 together, right? And we're going to do so using SCT register multimodal. So I see that Hosea has a question if I can share the command to register the MT on the native T2. Sure, I can do that. It's going to be this command. Running it here. All right, so let's copy this thing, paste it. So I'm going to explain a little bit the rationale for the different parameters. So input is the MT0, destination is the MT1. So we want to bring the MT0 to the MT1. We're using the segmentation of the MT1 only for the purpose of the QC, because you're going to see that the QC report centers on the spinal cord. And in order to know where the spinal cord is, then we need the segmentation, right? But we are not using the segmentation to register the T1 to register the MT0 on the MT1. We're using the mask. We're still using the mask because it's useful for a precise registration. And then the parameters we're using here are so only one step. We're using the image, and we're using the algorithm slice-reg, which is a regularized, X and Y translation, slice-wise, and we're using the cross-correlation matrix because these are two images with different contrasts. Right? We're using the spline interpolation in the end because we want to compute some quantitative metrics, so we want to be precise in our signal quantization, and the QC report. Let's look at the results, refresh. So that's our MT1, and that's our MT0. You see those two are pretty well registered. And once they are registered, what else we want to do? There we want to compute MTR, which is, essentially the ratio between the MT0 and the MT1 image. That gives the magnetization transfer ratio. So double click, copy, paste. It's a fairly fast command. It created a new file here called MTR, you see. And that's your MT ratio everywhere in your volume. Okay. Any questions so far? Okay. So before going further with the yes, we have a question.
Just so you know, there was a question from Gabriela that was asked a few minutes ago and got a bit lost in the message.
Yes, thank you. Thank you. This is going back to DeepSeg. Is there going to be a version of Seg lumbar SC that can be used for MFFE images? The website says currently it can only be used for T2-weighted images. Okay. Right. So I have two answers to that. One answer is that yes, there's going to be an updated version of SET DeepSeg GM. There's going to be a new version of that Niels Serre, I think Niels Serre is on the call, a PhD student in the lab, is going to work on. And I also encourage you to look at the current contrast agnostic model that might give you, satisfactory results in the lumbar spinal cord, even if you have MFFE images, which are essentially multi-echo gradient images. And then, send us some feedback. It would be appreciated to see how it works for you. And then, if we get feedback, then we can always improve our methods to make it work for your data. Good. Speaking of lumbar spinal cord, how do you register the lumbar region to the PAM50 template? So that's something we've already discussed before, but there are a few slides on it. So this is red sticker, so I'm not going to do it, but this is something I invite you to try at home. We have in the course material a lumbar T2, which looks like this. Okay. And then it shows how we can segment that lumbar part of the spinal cord. I'll give you this. And then the key here is to select the right landmarks. So you can use landmarks with only one label, label 17, which corresponds to the T9, T10 disc. Okay. It will correctly align that disc, but it might misalign the conus medullaris, right? Because it's not taken into account. So if you have multiple subjects with different sizes, you might have some issues there. You could use alternatively another label, the label 60, which corresponds to the conus medullaris. So you would have to manually identify it, each of your subject, and then use that in your SCT register to template syntax. Okay. So that has the advantage of making sure that your conus medullaris is aligned in all your subjects, but your discs might not be aligned properly. Alternatively, you can use two labels, one of the conus medullaris and one of the disc, and that will scale the image in the superior inferior direction such that your disc and your conus medullaris are aligned in all your subjects. So you see there are different flavors that could be used. And again, as always, it depends on your underlying hypothesis, the kind of analysis you want to run, the kind of questions, scientific questions you want to ask, and so on. Right. Again, if this is a bit overwhelming, feel free to reach out on the SCT forum. So this is more details on how to create those landmarks, and then the syntax specific to the lumbar spinal cord. All right, end of the day. Well, we're still going to spend a few minutes together, because we are doing well with time. So my suggestion is that we're going to come back to this in let's say 20 minutes, 30 minutes. But I think we can start the first section of the day 2 material, so that tomorrow we'll have more time. We won't be in a rush if we want to discuss, or if we want to go back to if you have more questions and things like that. All right, so we're going to talk about the gray matter segmentation now. A bit of theory. So historically, the first gray matter segmentation method that was introduced was based on a study by the Vanderbilt group, and it was based on an atlas, on a dictionary of different gray matter spinal cords. And there was a study that was implemented in SCT by Sarah Dupont, a former intern student. And that method was then replaced by SCT DeepSeg GM, which, as the name indicates, is based on deep learning. And that was published by Christian Perron in this article. So that former method is no more available, but nowadays this is the default methods for computing the gray matter. As I mentioned, we are working on a new version of the gray matter segmentation. It's not ready yet, but at some point we will likely replace this one with a new one. So for now, we're going to use SCT DeepSeg GM. So hands on the keyboards, we're going to try this together. And we're going to go to another folder that has some T2 star images of the spinal cord, again, on the same subject. So cd./T2s, that stands for T2 star. And there is this image in there. Let's see how it looks like. This is the folder, and this is image. So you see there is a nice, nice looking gray matter that we're going to segment. And we're going to use that syntax, copy and paste. All right. And while we run the full spinal cord segmentation, we're going to look at the QC. So I'm going to triple click, copy, paste. Okay. So while this is running, we're going to look at our QC. So QC report, refresh. And you see now a new file that is appearing here is the T2S, stands for T2 star, with my gray matter segmentation, right? So again, you can toggle on and off to verify, to make sure that your segmentation is looks good. And if it does not, for example, here, we have some weird spots, then you can manually correct them. And I'm going to show you later in the course, likely tomorrow, how you can correct for this. You see that the segmentation of the cord on the T2 star image is also finished. So let's refresh this one. And there is now a new line with DeepSeg, SAT DeepSeg for the spinal cord. So now you have this, the segmentation of the spinal cord and of the gray matter. And what are we going to do with those two? Well, we're going to subtract them. We're going to subtract the gray matter segmentation from the cord segmentation in order to obtain the white matter segmentation. Okay? And for that, we are going to use a new command that we are introducing here, SCT math, which is a copy of the FSL math from the FSL suite. It does the same thing. So triple click, copy and paste. It's a pretty fast calculus. So that's my T2 star. That's my segmentation of the cord, gray matter, and white matter, which is, again, cord minus gray matter. All right. So you can use that white and gray matter segmentation for different purposes. One purpose is to compute cross-sectional area of the gray matter and of the white matter. So for that, we're going to use the same syntax as Sandrine introduced when we were computing the CSA of the spinal cord. But now we're going to input the gray matter segmentation instead of the spinal cord. And we're going to call the output CSA underscore GM for gray matter. We're going to compute that for each slice individually. And we are not going to correct for the angle of the slice with respect to the angle of the spinal cord, because we don't want to introduce any error related to the estimation of the spinal cord center line. And the assumption that we're making here is that the spinal cord was, fairly orthogonal to the slices that were acquired for the T2 star scans. Copy, pasting, and same for the white matter. Copy, pasting. Okay, and so you see now I have two additional files, CSV files, one for the gray matter and one for the white matter. And if you open it, then you can see, for each slice, you have the mean. Obviously, the slice 0 has nothing because we didn't have any segmentation, and it looks pretty unreliable. You remember the little spots that we had that corresponds to like an unreliable value. But then the gray matter that was properly segmented gives you a cross-sectional area that makes more sense. It's in millimeter squared, right? So you have that for the gray matter and for the white matter. Any question? So you could use that segmentation to compute cross-sectional area. You can also use that segmentation to extract signal intensity within the gray matter. It's a red sticker, but I'm going to do it anyway because we have time. Okay, and then we can create it. This thing, T2S value. And so this shows the T2 star weighted value between slice 2 and 12. And that's the average T2 star value. And then you can do the same for the white matter. And it gives another result. Ah, yeah, it appends. Okay, you see there is a flag here that says append, which means that the value will be appended on the same file. So now I have two rows, one that corresponds to the white matter and one that corresponds to the gray matter. So signal in the white matter is 1300 something, and signal in the gray matter is almost 1500, right? And then you can use, for example, the ratio between the two to compute some, markers of, demyelination, if you think that this is relevant for your particular study. All right. Another thing you can do with your gray matter segmentation is that you can also account for it during the PAM50 registration, right? Because you remember when we did our PAM50 registration, we only used the contour of the spinal cord and we ignored what's inside the spinal cord. But now that you have the information of the gray matter contour, you can also rely on that to register your image to the PAM50 template. Because the PAM50 template has the information of the white matter. And now your subject also has the information of the white matter, outer contour and inner contour, right? So we're gonna see together now how we can tweak the SCT register to template command to account for the white matter. And this is a bit of a, an indirect path to register our metric to the PAM50 template by using the gray matter information that we get from the T2 star image or any other image where we can see the gray matter properly. So you see that this syntax gets a little more complicated now. It's like 6 lines of code. So we're going to try to understand that together. The input, so we're going to use SCT register multimodal as we did before, right? Because we are using an initial warping field, which is the PAM50 to the T2 template. So that doesn't change. Still have that. So our input will be the PAM50, but in this case, we're using the PAM50 T2 star, T2S, because this one shows a contrast between the white and the gray matter. And the input segmentation will be the PAM50 white matter and not the cord anymore. And the destination will be T2 star and the white matter segmentation on the T2 star. Initial warping field, as I said before, is the same, right? So we rely on the one from the T2. We also have an inverse warping field. That's how we're going to name our outputs, warping and inverse warping fields. And we have some, custom parameters that we know work well in this particular case. So let's copy all that, paste it, and then cross our fingers that it's going to work properly.
Meanwhile, I think we have a question from the chat.
Ah, yes, thank you. How good the segmentation should be to incorporate gray matter segmentation? Usually the merge images do not look very good for every slice, unfortunately. Excellent question and excellent answer. I think the answer is in the question. Well, as a matter of fact, I'm gonna have a slide, like in a couple of slides, talking about the fact that you really need a fairly good segmentation. There's no clear answer to how good the segmentation could be, because good is a fairly subjective adjective that depends on the kind of images, the kind of scientific questions you want to address, the working resolution of your image, the contrast. So it depends on many aspects. There is no simple recipe and simple answer to that. But it's good to ask the question because it makes you aware of how important it is. And my suggestion would be that to try with with the segmentation and without the segmentation of the gray matter, and then register to the PAM50 register a metric where you see the gray matter, and then you can decide whether the registration is better when you have the gray matter segmentation versus when you don't have it. To give you an example, the empty image that we registered here, that empty image does show the gray matter a little bit, right? So by overlapping the registered white matter, you can have a sense of how good that white matter is for this particular subject, right? So I would encourage you to compare this warped white matter versus a pipeline where you would use the gray matter from the T2 star or from like another series that shows the gray matter. That's something we can do together, actually. So yeah, it finished. This syntax is finished. So I'm going to run that, and then I'm going to look at the QC in the meantime. So now we are warping the object on the T2 star. Looking at the QC, refresh. So what you see here is the PAM50 T2 star registered on your image, on your T2 star of the subject, right? So what you can appreciate is if you look at the gray matter inside, then the gray matter is very well aligned with the PAM50 template. I know it might be difficult to see on your screen, so I'm just going to make it slightly bigger. There are some bugs that are happening here. Right. So that's, It's definitely better than what we had before in terms of gray matter. But as Merve said, it's not always the solution. One question from Gerardo, if you could use the PAM50 for pediatric data. Well, so it depends, first of all, how young, if we're talking 1 or 2 years old versus, 15 years old. I would say 15 is, much more reasonable. We do have an ongoing study that is led by Samuel Saintonge. I don't know if she's still in the Zoom. Let's see, Samuel here. Yeah, maybe she's not here anymore. But a student of Benjamin Delanier is working on multiple pediatric templates, and this is actually a work that is going to be presented at the next ISMRM in 2025. There's going to be a publication also out of that. But short answer is that yes, you can use it. Keeping in mind that there might be some limitations in terms of the shape of the gray matter that might not be adapted to pediatric population. But the scaling should be done fairly well, even if the spinal cord is much smaller. Okay, so the warping is done, so we can refresh. And then now what you're looking at is the warped white matter on the spinal cord. So you see that the registration is not perfect when it comes down to the spinal cord, but the gray matter is, gray matter shape is better. Still not perfect, I recognize, especially here, but that's something that could be accounted for by tweaking the parameters a little bit. Right? Okay, so now what we've done is this. We've estimated the warping field to go from the template to the T2 star, but we still need to do that. We still need to register the PAM50 to the MT using the information from the T2 star. Okay, so it's a pretty complex step, so bear with me. Again, this course is recorded. You can go back in time and play and pause at your own rhythm. So we're going to go back to our MT image, cd.mt, and then we're gonna run that command. I'm gonna run it, and then I'm going to explain to you what it does. Input is what? It's the PAM50 T2. Okay, so same as before. Segmentation is what? PAM50 spinal cord, same as before. Destination, same as before. We want to register it to the MT and the segmentation of the MT. That is the same, pretty much. But there's one thing that is different. It's the init warp. Because we are not using the init warp of the T2. We're using the init warp of the T2 star that we just calculated. And this one incorporates information of the gray matter shape. Right? And we're going to do a warped template. And then we will be able to compare, so to basically answer Merve's question, how good that is. How does it compare to the situation where we do not account for the gray matter shape? So there's one thing to consider here that might be a bit confusing to you. Is that we are, we computed an initial warping field that accounts for the gray matter shape, okay? But we're doing also other steps because this initial warping field is, used as at step 0. But then we have subsequent steps, step 1, step 2, that further modify the warping field. So we're kind of, modifying the gray matter shape specific transformation using, B-spline-Sin algorithm that is known to have very high degree of freedom, and that can further, counteract the effect of accounting for the gray matter shape. And that is why you need to be extremely careful with this choice of parameters. If you put too many iterations, then you will defeat the purpose of using that initial warping field. If you don't use enough iterations, then your registration will not be satisfactory because your registration will not adapt well to your MT image. So there's a thin line here. And again, it takes practice, experience, intuitions to get to the right parameters. So the warping is done. Let's compare our results with and without accounting for the gray matter shape. So as a refresher, here this is the result when we did the when we used the initial warping field from the T2. And this is the result when we use the initial warping field from the gray matter. So I'm going to make it a bit bigger and then alternate between the two. So with gray matter, oops, sorry. Where was I? Yes, this one. Okay, I guess it just changes one. Yeah, I cannot really zoom. That's a problem. I'm going to remove that. Yeah, that's a bit of a mess. Okay. So with the gray matter, without the gray matter, without. So you can see that here, for example, the gray matter shape is more personalized to this particular subject, whereas here it conforms more to the probabilistic gray matter shape of the PAN50. You see? It looks a bit better, but again, it looks better because we have a good gray matter segmentation. If you have a bad gray matter segmentation, then you can actually do more harm than good in your registration. So it's very important that you are aware of this limitation and that you choose carefully the way you want to register to template and the parameters that that you decide to use. Any question? Can take so it's 1:50 p.m. now in Montreal, so we're gonna keep that for tomorrow. So tomorrow we're gonna start at this this section number 9. But let's take a few minutes to chat. Open your mic, open your video. Oh yes,
There's also the poll.
Yes, thank you, Mathieu. Yeah, coming back to this guy here. We set up a very simple short poll that looks like this. So if you go back to slide 106, you will find the link to the poll. And you see these are very basic questions that will help us, tweak and change some of the design and features of SCT so that it's more useful to you. So please take a few minutes to answer it. And then in the meantime, I can also take some questions. So we have one question from Frederic. So currently I use SCT for the segmentation of the spinal cord for gray matter and white matter to apply microstructural diffusion models to spinal cords. The registration with FSL, but for the registration with FSL ants. I'm not sure I understand that. This is because my supervisor. Yeah. Maybe Frederic, would you like to open your mic and explain further? Because I'm not sure I understand. What you're
Sure.
trying to
So
do.
I can elaborate on it. It's because I'm working in a group where we try to apply the model to spinal cord. Of course, there are some issues with the size of the spinal cord compared to brain. And I guess my question is that when you are in a group where you use different tools for these images, you know, for
Yeah.
registering data to warp data to different spaces and stuff like that. There are,
Yeah.
you know, certain people has certain habits of what they use, and I tried to use the spinal cord toolbox, and in the end we decided on going with the registration tools of FSL. And I just I didn't have the arguments because more experienced scientists than me are used to do something, and I can't really convince them otherwise. But maybe you had something to.
So a couple of our, so what I understand is that you like what you would like to do, or like what you have in mind is to use SCT for the registration as well, as opposed to
Exactly, yeah. So I have my T2 star image, and I need to warp it, and I need to register it to DVI images, really.
So there are a couple of things to consider here. First, SCT incorporates some of the functions from ants. Okay, so it's possible to do exactly the same registration result using ants as if you are using SCT. Okay. That being said, there are some features that are implemented in SCT that calls ants that are spinal cord specific. One of them is the regularization or like is the constraint of the deformation along the axial plane. Because usually for when you want to register brains, this is done, in an isotropic way. Whereas for spinal cord segmentation, for spinal cord registration, we want to, in most cases, you want to have a good axial, refinement of the registration. And for this, you need to restrict the deformation in the Z dimension. This is what is let's see the register multimodal. This is what is corresponding to that particular field here. The subfield deformation, which is completely hidden, in this massive amount of parameters. But this is exactly what this does. You see that their default is 110, which is not the default in ants, and is not the default in FSL. I mean, FSL is using other algorithms, but in the end, these are also, similarly, highly dimensional, high degree of freedom, nonlinear deformation. But the point is that this is very specific to spinal cord, and that could help in your case if you want to register spinal cords. Another argument in favor of SCT is that there are other algorithms that have been developed specifically for the cord. One is the column-wise. If you have, very, very like a large amount of deformations and you want to rely on the segmentation. Center mass rot is something that does not exist in FSL or ANTS. There's a deep learning approach also that I did not mention, but it could potentially be relevant in your case. And what else? I think there's also, yeah, so this is also specific to SCT. But you see, there are just like a couple of very specific tools that that we've developed for the application. But that being said, maybe you could get as satisfactory as SCT results with ANTS and or with FSL. The best is to try. If you have some example images that you would like us to give to give it a try, to optimize some parameters, feel free to post them on the forum, and we could spend a few minutes, trying to optimize. Because in the end, at the end of the day, I think registration is one of the most complex tasks in computer vision, and it does not come down to, oh, is FSL better than SCT? It really comes down to amongst the hundreds of parameters that are part of each algorithm that are part of FSL and SCT, which are the ones to use, how to use them, and that decision depends heavily on your images, right? So I haven't seen your images, so I can't really comment on that. But it's a difficult process. And again, happy to assist if you want to share some images.
That's great. Thank you so much. Yeah, it is very complex, and I'm not very experienced. So, but I guess I'll use the forum to get some help, and we could discuss it. That will be very nice.
Sounds good.
Thank
Then
you.
there's another question from Marco. Is it possible to register two images, same modality by same modality? I assume you mean same contrast, right? Like 2T1, for example, recorded at two consecutive time points and use the warping field generated from registering them to compute spinal cord atrophy. Yeah, I think you're thinking probably in terms of the like VBM type of approach, huh?
Yeah, correct.
So it is, well, it is possible. Many things are possible. Questions now is, how relevant and correct that would be. I could see, yeah, I think that makes sense. I think that's possible. The real question, I think, is, how correct would the registration be? Because you want this registration to be excellent and to surpass the precision and the accuracy that you would get from the spiral cord segmentation. So I think that's really the question at stake here.
Okay.
And
So
yeah, go ahead.
in the toolbox, there is any like tool to measure atrophy? There is not like the difference between cross-sectional areas?
I don't think there is. I don't think there is. No, I don't think there is. You could think about it.
Okay, thanks.
Yeah. Feel free to open that discussion on the forum, and then we can continue on that. It's yeah, it's a good idea. Yeah.
Thank you.
Any other questions?
Yes, this is Fauzia.
Hi.
Hi, thank you for organizing this tutorial workshop. So my question is, we are trying to extract morphometrics from clinical based MRI, not and the resolution of the images is anisotropic. It's 0.8 by 0.8 by 3 millimeter. What advice can you give us for a T2 image?
For what is the, are you trying to compute cross-sectional area?
Yes.
Okay. And the orientation is axial, I assume?
Yes. I can really share the data on the forum. So we've done the isotropic, which is 0.8 by 0.8 by 0.8 for the same patient, and then we're trying to compare with their clinical-based MRI to see if we can actually be able to do the morphometrics on those images.
Sure.
But segmentation was a problem.
Ah,
yes.
I see. Have you tried with a recent contrast agnostic model?
No, not yet.
So that would be my first advice.
Okay.
Try with this new model, see what you get.
Okay.
In the end, unfortunately, if the images are, image quality is too low and we are not able to, even with our naked eyes, to distinguish what's the border of the spinal cord, then, there's no way an algorithm can do it in a reliable way.
Okay.
It always comes down to the quality of the image. If the quality is sufficient, then, the fact that it's anisotropic shouldn't be too much of a problem.
Okay. Yeah, I'll give it a try and then maybe send a sample on the forum to see if.
Cool. Sounds good.
Thank you.
We have a question from Paul S. I don't know if it's Paul Summers, but yes, covered tomorrow. Exactly. Very exciting. It's a new feature. Oh, yes, Paul. Hi, Paul. How are you? All right. So it's 2:05. We are 5 minutes late, but it's been a good session. We covered a lot, so I think it's time now for you to get some sleep and to consolidate all that knowledge to be ready for tomorrow for more fun. So thanks everyone for attending, and then see you tomorrow, same time. Bye bye.
Thanks.
Yeah, just
Record in progress. Okay. So gonna pull the chat. Excellent. Welcome back, everyone, for this second and last day of the Spinal Cord Toolbox course. So maybe before we start, do we have any questions? Would you like me to clarify something, from all the material that we covered yesterday, which is pretty dense, I realize. If you cannot think of something, or like if if your questions comes up later, feel free to ask anyway, even if it's unrelated to the thing that we are covering at the moment. So I'm gonna continue on the slide. So we just finished slide 8, and remember we were showing how we can improve the accuracy of atlas-based registration, and more specifically taking into account the shape of the gray matter to better match the gray matter contour of the PAM50 template. And next, what we're gonna do is we're gonna see how we can use this registration to the template to extract quantitative metrics from DTI, magnetization transfer, fMRI, activation blobs, and things like that. So typically, using the atlas of the white matter to extract information within specific tracts of the white matter. And if you're doing fMRI, it could also be extracted inside the gray matter. A little bit of theory. So if you want to do atlas-based analysis, the same way, some of you might have done that already in the brain, is that you need an atlas. So in Spinal Toolbox, there is a digitized version of the Gray's Anatomy white matter atlas that is merged with the PAM50 template. And so for more information, you can see you can look at this article from Levy et al. So there is 30 white matter tracts, 6 gray matter regions, and then the surrounding cerebrospinal fluid. So I'm going to talk about something called partial volume. How many of you have heard of the concept of partial volume effect here? Can raise your hands. Yeah. Okay, see a couple of hands. That's good. So partial volume is bad. That's your enemy. You want to, mitigate it by acquiring, with a, small voxels as small as possible. This is why you want, high resolution in your scans, but you can never obtain, like nanometer scale with your MRIs, right? You're always gonna get voxels that are on the scale of, let's say 1 millimeter. And so what happens if you're looking at the tiny structure, such as the spinal cord, is that you're basically, discretizing your regions of interest. If you look at, for example, the spinal cord here, this is like a zoom on a voxel here. That's what you see in the red square. I'm zooming in this voxel, and inside that voxel, you have different tissues. You have the cerebrospinal fluid, and then you have next to it the spinal cord. Obviously, this is a very simplified model because you also have the dura mater, PL surface, and so on. But let's say that the signal in the spinal cord is, let's say, 50, and the MRI signal in the CSF is 0. Let's say you're using inversion recovery sequence that that nulls the signal in the CSF. Well, because what you get with your MR image is only this voxel that includes those two structures, the signal in that voxel will be 25, like an average between the two, right? If the occupancy of the spinal cord and the CSF is 50% each. So, that's exactly what partial volume is. You're losing information. You cannot properly disentangle what's the contribution from, the spinal cord and from the CSF. Well, there are some techniques to, try to disentangle this and to tease apart the contributions from the different compartments. Well, one very simple approach is essentially what we do when we have a binary region of interest. Let's say when you have a binary mask of the spinal cord, you could average all your the signal in all your voxels, gives you a value, but it does not account for partial volume effect. One way to mitigate partial volume effect is to do a weighted average. So in this case, if you have a soft mask instead of a binary mask, so let's say that in the vicinity of the spinal cord, the mask, instead of being 1, it would be, for example, 0.5 or 0.25, right? So in this case, you would weight the MR signal depending on how confident you are that the signal is coming from the spinal cord, right? So that's called the weighted average. And then there are two other approaches, maximum likelihood and maximum a posteriori, which originate from Bayesian theories, and they they basically make some assumptions that inside the spinal cord, the signal is the same, or inside your region of interest, the signal is homogeneous, let's say 50-50 everywhere. Using that a priori, you could solve a mathematical equation, where essentially X is the voxel covering all your regions, that includes the spinal cord, but that also includes the surrounding cerebrospinal fluid and gray matter and so on. And when you have this information of what's the fractional contribution of each tissue in each voxel, then you can recover the actual signal in the region of interest, right? And maximum a posteriori is a similar version of the maximum likelihood. I won't go too much into details. These are more explained in the Levy et al. Paper, but I'm just going to show you how that's helpful if you're looking at very small tracts. For example, if you're looking at for example, the tracts like the lateral reticulospinal tract, which is a very tiny tract on the side here. In green, it's the maximum likelihood approach. In blue, it's the weighted average approach. And in red, it's the maximum a posteriori approach. So you see that you're making less errors if you're using methods such as the maximum a posteriori compared to the weighted average, which is jumping all over the place. And that's because there is noise in your data, and also because of this partial volume. So there are two papers that describe this phenomenon with simulations that I invite you to look at if you're interested. But this little part of theory is really to make you aware that there are different algorithms in Spinal Cord Toolbox to extract your DTI or MT signal. So it's good that you're aware of those different algorithms so you can carefully choose the one that is more adapted to your data. Any question on this so far? Okay, so if there's no question, then we're gonna make this into practice.
Sorry, I do have a question.
Of course.
Can you hear me?
Yes, very well.
Yes. So we use the same command line like SCT extract metric for gray matter, white matter signal. That intensity when yes, the intensity when we're trying to calculate the intensity of the gray matter. In that situation, what do we use among this algorithm?
So that's exactly why I didn't want to tell you, which one you should use. Well, although it says recommended here, so I guess this is recommended in some ways. But it really depends on your data, the amount of noise, the quality of the mask, how reliable it is. So that's why I prefer that you understand how these algorithms work instead of telling you should use this or that. See what I mean?
Yes, thank you.
I know this is really not a satisfactory answer, on the contrary. But I think I invite you to maybe look at these articles, those two articles, because the more recent one also has additional simulations with respect to the resolution of the MRI scans. Let's say if you have a scan that is 1 millimeter isotropic versus 2 millimeter isotropic, what is the method that you should use? And that article addresses it. And there is really no simple answer to that, because it depends on the size of the region you're interested in, on the amount of noise. For example, what you see on this red square here is that the maximum likelihood and the maximum a posteriori pretty much give you the same answer, right? So I would tell you, well, it doesn't really matter here. And you are talking about the gray matter and the white matter. Well, these are even bigger regions. So, you know, personally, I would go with the maximum likelihood approach if you were to extract the white and the gray matter. But, you know, maybe the amount of noise is all over the place, and maybe that would not be robust enough. So that's, you know, we're gonna talk about this a little further in those slides. So what you're seeing here is a file that I already opened yesterday. I don't know if you remember, but when we used the SCT warp template, it generated some files under, you know, the label atlas folder. So let's say if we go to the empty folder where we run warp templates, it created this label. Under this label, there is atlas and template, which are the PAM50 objects. And this atlas is the white matter atlas in the space of the empty scan. And there is also an info label file, which is, you know, the same as the one that I'm showing here. And it shows that, you know, it basically refers to all the different files. So the PAM50 atlas 00, which is this one, corresponds to the left fasciculus gracilis, okay? What you're seeing below here is the combined labels. And it says that the spinal cord is composed of the labels 0 to 35, right? The white matter is composed of the labels 0 to 29. And this information here is used with the algorithm maximum a posteriori. Because the maximum a posteriori algorithm, if I go back to this slide here, uses some prior information from the average value in the CSF, white matter, and gray matter. That's what this beta nut is here, okay? So that's why it's important to, you know, to not touch these numbers here, these files, because it's using this with the algorithm maximum a posteriori. Now, if you'd like to, you know, customize this file, and then let's say you want to add, you know, the left part of the cord or the right part of the cord. This is what that was a request a couple of years ago from John Brooks in the UK, and then, you know, he posted that on the forum. Actually, the link to the forum is here, and so you see that you have the flexibility to add. So do not change this line here. That you don't touch, but what you can touch is adding some numbers afterwards. You can add 56 and then left hemichord, and then add the labels that corresponds to the left part of the cord. Then you can add 57, that would correspond to the right part of the cord, right? So you can personalize depending on your study, we know what are the regions that you want to quantify your quantitative MRI signal from. Does that make sense? Okay, so now that you're familiar with, you know, this concept of info label, we can use it. So open your terminal. You need to go. I don't know if you know, switched off your PC or your Mac, but you need to go back to the correct directory. So for example, I reopened my terminal, but I'm not at the right location, right? If I write this, it's gonna fail. Gonna fail because I don't have that MTR file here located, right? So let's go to the correct folder. In my case, this is my home folder, Julian. That's where I am, PWD. So I need to go to the SCT course data, SCT course data tab so that it's faster, and then single subject, data, and then empty. Good. So that's the right location. Now I can, so up arrow, I don't know if you know this trick. If you press on the up arrow, it shows what you wrote in the history of your terminal. And now I press enter. It loads the matrix, which is the MTR. And then it's using the label Atlas, this folder here. It's recognizing that there is an info label. It's using the method maximum a posteriori. And then the -L means that we want to extract the metric in the label number 51. What does the label 51 correspond to? Well, you can list, that's the tip here, you can list all the available labels by typing SCT extract metric list labels. So let's do this. You see, 51 corresponds to the white matter. So what we did here is that we extracted MTR inside the white matter. And the output is the file mtr in white matter.csv. Okay. And that's the file that I'm seeing right there. Okay. So let's open it. And you see that there is a bunch of stuff. Oops. Can I zoom in? I can, okay, I can zoom in. So when the command was run, the version of SCT, the file name, mtr, that's fine. The slice, the name of the label, in this case, white matter. The size, so the size is important. It tells you the number of voxels in the fractional voxels, because it's not a binary mask, right? The white matter mask is not binary, which is why you have float numbers as opposed to integer. And the value of the maximum a posteriori signal estimate. So that's in English, it means that's the MTR inside the white matter. And you can see that, it's pretty stable. So that's nice, 55, 52, 53, 52, 50. And, to address your question, Fozia, when you were asking, what is the right method? Well, the right method has to do with the size of the mask that the metric was extracted from. In this case, this 52.5 here was estimated by running a statistics on, almost 71 voxels. 71 voxels is pretty good. If your mask was like 3 voxels, then you should be concerned. If your mask has like 1,000 voxels, then you're fine. That's a pretty robust statistics. Except if the inside your 1,000 voxel mask, you have some assumed heterogeneous signal coming from the MRI, right? Let's say if you have your white matter mask, and inside the white matter you have big lesions or like a strong artifact, like dropout artifact due to metal implants in the spinal cord injury population, for example. Well, in that case, your mask will include normal appearing white matter, but it will also include regions that is highly artifacted, right? So even though your mask would be big, the estimated extracted metric would be incorrect because it would not follow assumed Gaussian distribution inside your mask, right?
Okay, thank you.
So this is a very important concept. So I hope that everybody's following what I'm saying here. And the STD here corresponds to the standard deviation within the mask. Okay. Sounds good. What can we do next? Well, we can extract the white matter within the right and the left corticospinal tract. Which are they? Well, let's look at our list. Left lateral corticospinal tract, that's the label number 4. Right CST, that's the label number 5. Okay. And what we also want to do is to extract this within specific slices. 5 to 15. Okay, so let's do this. Double click, paste. Okay, so the output is this file here. You see that each row corresponds to a particular label. You have the size of the mask inside the lateral corticospinal tract. So you see that's funny because 60, is almost the same as, the 70 that we had before. Before we had the size of 70, but that 70 here, well, like 60, 70, corresponded to the size of the entire white matter for a single slice, okay? What we're doing here is different. We are looking at the size of the lateral corticospinal tract, which is much, much smaller than the white matter, but we are looking at it over 11 slices, 5 to 15, right? So the statistics is still fairly robust because you're averaging across different, I need to remove my thumb again. Okay. Okay. So, the statistics is still fairly robust because your mask is, big enough, which is why if you want to extract in very tiny tracts, I strongly recommend to do it over, large number of slices. Then maximum posteriori, about, 52 in the right and in the left corticospinal tract, which, it brings confidence because that's what we would assume in a healthy individual to have the same value in the right and in the left CST. And you can also specify the vertebral levels to extract your MTR from, because we also have access to the templates and to the vertebral levels of the PAN50. So to do that, you would input the MTR, same as before. That's same as before. Same as before. 53 is the dorsal column, as you can see here. Dorsal column is label 53, and this is the assemblage of the tract 0 to 3. And 0 to 3 are the fasciculus gracilis and the cuneatus, right? Left and right. And we're doing that over vertebral levels 2 to 4. And let's see what we get. Output is here. The C dorsal column. Going to zoom in. So now we are doing it over the slices 7 to 16, which corresponds to vertebral level 2 to 4. Label is dorsal column. You see the size is much bigger now because we are looking at, several slices over a region that is bigger than the lateral corticospinal tract. And maximum a posteriori value is about 53, right? So slightly higher, possibly because there is also a higher packing of axons in the dorsal column has like smaller axons that are much more dense, maybe higher myelin content, possibly. Okay, any question? Okay. So let's go to the next section, which has to do with diffusion-weighted MRI.
Sorry, I have a question.
Yes, of course, Fozia.
I'm asking because we are currently working on an MTR and your team have been very helpful. What I noticed from our own data, because we've collected them on a GE scanner, even from a healthy control, and when we take the MTR, when we average between level 2 and 3, it's still much lower than, I think the highest we've gotten was like 40 something, the percentage, the MTR.
Mm-hmm.
So I know there's one of your paper that you compared, the MTR was slightly different depending on the scanner. I don't know is that something that is acceptable? Because I've seen the numbers here, it's like in the 50s, right? This is a normal subject, so.
Well, I mean, you answered your very good questions by pointing out to the fact that depending on the sequence, not necessarily the scanner, but the sequence parameters, you get different MTR values. And that has to do with the fact that, as like the way MTR works is by, doing the ratio between two different images, one with and without the off-resonance pulse. First thing to consider is that this off-resonance pulse, could have a different frequency depending on the vendor. The shape of the pulse could be different. Could be sync pulse, a square pulse, depending on, or Gaussian pulse, depending on the vendor. That has an influence on the amount of magnetization that is sent to the spins before the imaging pulse. Another point, very important to consider also, is that you might sometimes use fat saturation in your images, and fat sat is essentially an off-resonance pulse in itself, right? So by adding fat sat, you're essentially then computing a ratio between two images that have already some off-resonance pulses, right? So your delta of MTR that you're looking for would be smaller than if you are not using an off-resonance pulse, like an off-resonance fat sat pulse. There is obviously also effects of the TR and the use of like flow reduction pulses, flow reduction gradients. Short answer is you'll get variability in the MTR. There is no single MTR value for the white matter, and this is exactly why people have been working for decades now to develop what they call QMT, which is quantitative MT value, which is not dependent on so some of these parameters. So I won't dive too much into details because this is out of the scope of this course, but if you want more details, feel free to post on the forum, and then I'll redirect you to some literature.
Okay, thank you so much.
Any other question? All right, so diffusion MRI, that's again a technique that we could spend hours discussing and describing, but very brief like pitch about what DMRI is. It's an MRI technique that is sensitive to the random movement of water molecules. As you know, with MRI, we image the hydrogen protons of the water, which is pretty much everywhere in the body, including in and outside of the axon bodies. And what's interesting with the water that is in the axons, in the vicinity of axons, is that because of the fibrous structure of these axons, similarly to if you were taking a celery or like like a leek or like all those kind of vegetables that are very fibrous. If you put those vegetables in the MRI and you run a diffusion MRI sequence, you will notice that there is, more diffusion, like the distance of diffusion for a set time is bigger along the fiber versus across the fiber, right? And that's exactly what's happening also in our axons. And the idea of MRI is to apply gradients, magnetic gradients, along this direction, but also along this direction and along other directions. And then if you apply a gradient along this direction, then the water molecule, the spins will essentially dephase much more than if the gradient was applied in this direction, because they move more along this direction versus this direction, right? So what you're measuring with diffusion MRI is essentially the phase, the phase of the spins, and that is translated in like a smaller magnitude signal if there is more diffusion. And that dephasing, or like that, phase related change in the magnitude signal can be modeled using what we call the mathematical tensor, which can be obtained using singular value decomposition. And it gives you, it's a 3D representation of the diffusion in space. So it's represented by a 3x3 matrix. And from that matrix, you can extract, some like aggregated description of the diffusion. One that is useful is the axial diffusivity, which describes, how much there is diffusion along the group of axons. Radial diffusivity, this one. Mean diffusivity, which is, kind of an average between the free and the radial and the axial. And the fractional anisotropy, which tells you how, how anisotropic your diffusion is. If you were to measure FA in the glass of water, then the FA would be almost 0. But if you measure FA in fibrous tissue, then the FA would tend towards 1. Right? So FA is always between 0 and 1. So, something that I said in the very beginning is that SCT is not really is more seen as a pre-processing package, right? So there's already a lot of diffusion MRI toolbox out there, MR-Trix, DiPi, many others that are extremely complex, actively maintained. So the goal is not to replace those. The goal is to only address some of the features that are not covered or like that, partly covered by other packages, or like that don't, that are not well covered for the spinal cord by the other packages. Okay, so hands-on on the keyboard, and then let's go to the DMRI folder, the cd.dot/DMRI, and then we go to this one. So what does this thing do? Or maybe, you know what? I'm always saying, let's look at the data. So let's look at the data. DMRI. Just going to open the data with Facialize. Just so you have a sense of, what the data is like. All right, so that's my DMRI series. The first one is what we call the B0, so there is no gradient applied. Okay, so it's essentially a T2-weighted scan, T2-weighted EPI. And you see there is a volume here because it's a 4D volume. And the fourth dimension corresponds to the time. So this one is another volume, and so on. And what you can see is that, there is almost no signal after the volume 0. And that's because here the diffusion gradient were applied. What's also funny is that depending on the volume, we can see the spinal cord, or we don't see it well. And that's because in this case, the gradient was applied orthogonally to the cord, whereas in this case, it was applied longitudinally to the cord. right? So superior inferior direction, because most of the fibers in the spinal cord are running in the superior inferior directions, right? You can also make a movie out of that if you like cinema and so on, right? Okay, so the data look pretty good. Not sure what's going on. Okay, I'm just gonna stop that. Data looks pretty good. I invite you to always, look at your data before you run the processing, to make sure that, the field of view covers the spinal cord. There is no weird artifacts and so on. Okay. So the first thing we're gonna do is we're gonna separate the B0 scans and the DWI scans. And we do that using the BVALs and the BVEX, which should come alongside your nifty DMRI scan, according to the, if you use DCM to NIIX to convert your images from DICOM to nifty, you're gonna get those files. And these are, it tells that the first volume is a B0, the second volume is, B-weighted with 800 second per millimeter square, and then the last 4 volumes are B0 scans. Okay. So it finished, it created, an average of the B0 scan, an average of the DWI scan, where we can see very well the spinal cord, right? So that's a beautiful scan for segmentation that we're gonna use. Next, we run DeepSeg to segment the spinal cord on the mean DWI scan. And, we use the contrast agnostic as we did yesterday. And after we do that, then we're going to use that segmentation to create a mask around the spinal cord center line. And you'll see, well, actually it's written here. We'll be using that mask in order to perform motion correction. So it's very similar to what we did with the empty scans. I don't know if you remember, we created a mask around the spinal cord to have, more accurate registration between the MT1 and the MT0. So, we do the same here. Create a mask. Or maybe before, let's look at our QC. So, purple click, copy paste. This is our QC. You see, I still have all the QC from yesterday, and it's just appending this to the end. And so that's our DWI mean scan with the segmentation. So it looks good. Now we can run, oops, that's this one. Now we can create the mask. That's going to have a size of 35 millimeter around the spinal cord center line. Right? So that's our mask here. So far, so good. Okay. So now we can run the motion correction, and that motion correction has a couple of features that were inspired by the article from Shu et al. in 2013. Some of the features include the outlier detection. If displacement is too large, then it would be considered as like an erroneous estimate of the motion. There's also slice reg, which is a regularized translation along X and Y, accounting for the fact that, because you're acquiring axial slices, we should not expect, a very strong motion in one of the slices compared to the neighboring slices. Right? So that helps motion correcting data, which are inherently very, very noisy. So double, triple click, copy and paste. Right. So if I go up a little bit, you see that it's identifying all the B0s and it's identifying all the DWI scans. And it can identify them because the input of the command includes the BVEC file. Right? which are the it indicates the orientation of the diffusion gradients. So it splits the data, it runs the motion correction on the B0, then on the DWI, then it combines all them together. And that's it. We also have a QC for the diffusion-weighted motion correction. So let's open the QC. You see, it's like a little GIF. I was also inspired by FSL that has a similar QC, similar HTML QC in their suite. Then you can basically compare the before and after motion. Well, fortunately, this subject did not move very much, you know, in the beginning. So you know, there is no big difference between the before and after. But if you have subjects with strong motions, that's where you can start appreciating the benefits of motion correction. And one thing I should add also is that because this process is a bit tricky and you're dealing with noisy data, sometimes you realize that you're going to have more motion after applying the motion correction algorithm, which is very counterintuitive, right? But that's just because you know you're applying an algorithm that might not be robust in some cases. So you might want to play with the mask, you might want to play with the you know, some of the parameters, the cost functions. If you'd like more insights and help, maybe that's a good idea to open an issue on the SCT forum and share your concerns with your own data, and then we can chip in and have a discussion. Sounds good? So yeah, maybe one thing to mention also that is in the presenter note at the very end is that one of the outputs of motion correction is the TSV file. You see this TSV MoCo params. It shows the mean square displacement for each volume 1, volume 2, volume 3, and so on. For each slice, but wait, doesn't make sense. Doesn't make sense that it shows it for each slice. I think that's wrong. Mathieu, I know you worked on this. Do you remember exactly why we're only showing? Because if we have several volume and several slices, that doesn't make sense to me. Okay.
I know we used to show absolute distance in the X and in the Y separately, but we decided that it makes more sense to show absolute distance. Actually, it wasn't absolute. There was positive and negative numbers. It was a
Oh, I think, yeah. Okay. Well, there is more information in this issue. So we should probably look more into that. I don't want to spend the entire course. Yeah, that's for so that's the sum across all the slices, right? So what we're showing is the displacement across all the slices for a given volume.
I think it's averaged. There's 1 over n in front.
Yeah. Yeah. Okay. So yeah, something to clarify. If you want the information for each slice, you would have that in the MoCo params X. And that's something that, especially if you do fMRI, you might need that for your regressors, right? So the fMRI MoCo is similar. We'll see that later, but you know the parameters for each slice along X and along Y would be in these files. In the interest of time, I'm not going to go too much more into details, but you see that the informations are there. And if you need more, more details, clarifications, then, we can discuss that in the SCT forum. Sounds good. Maybe a note, Mathieu, to clarify what's written here. All right, so now that we've corrected oh yeah, so there is a question from Ajay about the shearing effect, eddy current artifacts. Yeah, that's a very good question. So it's it is possible definitely to apply algorithms like top-up, for example, to account for eddy currents. A couple of thoughts. On that. And in fact, I think there is whole discussion on this.
I just sent a link to the discussion to the chat.
Oh, thank you. Great. Excellent. Thank you. That's the one I was thinking about. So there is, and Court is also part of this. That was a long time ago. Actually, not so long ago. But yeah, so Ajay, this is a very relevant question. There's no simple answer to that. Really no simple answer to that. But feel free to chip in with your thoughts, and then happy to continue the discussion on that. Okay, back to the course. What we're doing now is we are rerunning the segmentation, but now on the motion corrected mean DWI, right? Because we accept that this one is more precise compared to the non-motion corrected DWI average. We are running this one, and then we'll be able to compare with our QC. And then after we do this, we'll be able to register the PAM50 template to the DWI scan via the T2 T2 registration. Okay, so this is done. Okay, and then we're gonna look at the QC. Refresh. Segmentation looks good, but so you see, we can easily compare. You can easily compare the the motion corrected and so that's the non-motion corrected, and that's the motion corrected DWI. So you see they look pretty much the same, because again that subject did not move very much. But if you have a subject that moves a lot, then you're gonna see some more differences. All right, so next we want to register to the PAM50. So I'm going to copy, paste it. And then while this is running, I can explain to you all the different parameters. So the input is the PAM50. Now we're using the T1 contrast. And the reason we're using the T1 contrast is because the cord is bright, CSF is dark, and it looks very similar to the actual DMRI DWI scan, right? If you look at the DWI, which is this one, cord is bright, and then surrounding CSF is dark. So it's a similar contrast in the T1, which is why we like to use this one for registration. Input segmentation, same as before, PAM50 cord. Destination, motion corrected DWI scan. The destination segmentation, well, the segmentation that we just computed. Initial warp, very important. It's the, template to ANAT that we computed yesterday. And also the inverse warping field, so that we can also output the transformation to go from the DWI to the template. Okay. And these are our custom parameters, right? We are using the segmentation as a start. Center mass, we don't need the rotation because it's already accounted for by this one. Again, same subject, same position, pretty much. Step 2, we are using the segmentation and the B-spline scene algorithm. All right. So now we can look at the results. So refresh, register multimodal. And these are now that's our mean DWI, and this is the registered PAM50 T1 registered to the DWI. So it looks pretty good. Any question? Okay. Well, next, what you want to do ultimately is to compute quantitative MRI metrics, right? So what you need is, compute the diffusion tensor. Again, this could be done using other software, but we implement a very simple, DTI metric estimation using DIPI, via SCT. So that's it. The output, you see all those files, DTI, axial diffusivity, FA, MD, and RD. And then finally, we can extract metrics. And we're going to extract the FA inside the white matter. That's what the label 51 stands for, using the method maximum a posteriori between C2 and C5. And we want to have that pair level. So that means that I want my average FA at C2, at C3, at C4, and at C5. Okay, and it crashes because I did not run SCT warp template. So there's a problem in this. Oh, sorry about that. I forgot that one. There's no problem with the slide. There's a problem with me. But you see, that's part of a regular image like processing. It's very common to have errors, and what's very important when you see an error is to not freak out. The good strategy is that if there is a problem, then there's a solution somewhere. That's pretty much always the case with computer vision. So what it says here is that label Atlas does not exist. So your first reflex and strategy when you have an error is to try to understand what's the source of the error. So it does not exist, well, because it should exist. And how to make it exist? I know that this is by running the command SCT warp template. So what I'm trying to say here is that it's a little bit like cooking. When you prepare a dish, you can either follow the recipe, try to follow the recipe line by line, and it's never going to be the same as what a chef does, because the chef has a lot of little tricks that are not listed in the recipe, and also the recipe varies depending on the temperature of the room, depending on the quality of your ingredients, depending on the humidity level and things like that. So you always need to, understand the kind of processing you're doing, the kind of data you're working with, as opposed to, just following a script line by line and applying it to your data. All right, so we warped the template. So then I should be able to run that command. So let's see. Okay, so now we can open the CSV file to see what's in there. This one. And I have fractional anisotropy at level C5 that corresponds to the slices 1 to 3. So I did not have to specify those. They are automatically found using the the vertebral labeling of the PAM50. And then at level C5, I have the white matter that has a size of 225 voxels, and the average FA is 0.79. Same for C4. The FA is 0.8, 0.8, and 0.8. Right? And you have the standard deviation within each region of interest. Any question? Okay, so there is no question. We're going to go to another popular quantitative MRI technique, functional MRI. So very briefly, fMRI is a technique that can measure brain or spinal cord activity. It's horrible. It's written brain activity in my slides. I need to change that immediately. Let's see, what page is that?
Fixed.
Brain. Thank you, Jan. Brain and spinal cord activity, or brainstem. Like, you know what? Let's just be spinal cord activity in the central nervous system by detecting, changes in the blood oxygenation and flow. So it's really, what's important to understand is that this so-called BOLD signal, blood oxygen level dependent, is an indirect way to measure neuronal activity. Right? It's different from inputting electrodes and measuring the synaptic pre or post synaptic activity. You're really measuring the the metabolic response to a neuronal activation. So BOLD signal is confounded by many parameters, but nevertheless, it's a very popular and in some cases good surrogate of neuronal activity. And typically, we like to use gradient echo EPI to measure the BOLD signal because they are gradient echo because they are T2 star weighted. And what we are after is changes of blood oxygenation or, being sensitive to changes in paramagnetic concentrations. And that is seen using, sequences that are sensitive to changes in susceptibility profiles, such as gradient echo image, as opposed to spin echo image. And echoplanar images, just because these are very fast to acquire. Right? You get a volume in 1 second, approximately. All right. So this is a very busy slide with a lot of things to do. So let's go line by line together. Going back to my terminal, first thing to do is to go to the fMRI section, fMRI folder. Okay, I'm there. Let's see what's in there. Only one scan, fMRI. Again, it's good to look at the data. So let's look at the data. Personalize. Right. Okay. Let's change the intensity. These are my different slices. So you see it's a bit blurry. It's a bit artifacted. That's typical of a gradient echo API. See, there is some CSF flow disappearance depending on the slice. You see the CSF there, but you don't see it there, right? Because of there is no flow compensation. There is susceptibility artifact because of the disk that is close to the region of interest. So it's a very ugly looking type of MRI. But nevertheless, you need to deal with that when you do fMRI. Okay, so these are the different volumes. See? I think there is a bug in my that's the second bug in 2 days that I observed with like with Facelize. Okay, so we're just going to do this. So it's good to look at the movie of your fMRI time series to have a sense of of the motion, of how the signal changes across the different volumes. So what you're seeing here, for example, is that depending on the volume, sometimes you see the CSF and sometimes you don't, right? That's because CSF flows, and then depending on where you are in the cardiac cycle, and you will see it or you won't see it. Okay. So not so much motion. We can see the chord. First thing we're going to do is average all the time series across time. And then we're going to use the mean fMRI scan to do some mask creation and then motion correction. So we could right away segment the chord, but in this case, I don't want to do that because I want to show you another way to create a mask and to do motion correction. So it's really for the purpose of this course. But if you process your own data, you don't need to do what I'm going to do here. I just want to show you that we can use register multimodal by taking the T2 segmentation, right? And bringing it to the space of the mean fMRI time series. But I'm not going to do any fancy algorithm to do the registration. I am just going to use the Q form that I talked about yesterday that defines the region of the field of view in the absolute coordinate system. And by using the Q form of the T2 and of the fMRI, then we can figure out a matrix like a transformation that brings the T2 segmentation to the fMRI mean. So there is no estimation, there is no cost function. It's a very simple transformation that is applied. So you can see that as a resampling. We are resampling the T2 segmentation in the space of the fMRI mean. And we can see what's the result of that. So I'm going to run that command. So again, this is not a registration per se, but it's a resampling, right? So in red, I'm going to put the registered T2 seg. And then I'm going to toggle back and forth. So you see, it looks like I segmented my fMRI scan, but in fact, I did not. The only thing I did is I took the segmentation of my T2 and I put it in the space of the fMRI. So, it looks pretty good, but you see that this one is not perfect. For example, there is a shift because of susceptibility artifact, because the subject might have moved between the T2 and the fMRI, right? But so the reason I did it is because it's already a good start, and I'm going to use that to create a mask, and that mask will be used to run motion correction. Does that make sense? So let's do this. We're going to create a mask. Okay. Then we can look at the mask. I'm going to open it. And it's mask fMRI. And then we can put it in yellow with some opacity. Right? So I just created a mask around that registered T2 weighted scan. Sorry, T2 weighted segmentation. And now we're going to use that mask to say, hey, I want to motion correct inside the mask. Okay. So that's what we're going to do now. You see it looks across all the different volumes here. I have 50 volumes in my fMRI time series. And by the way, if you want to play with the fMRI motion correction, there are different algorithms that are, that you can tweak. If I open another terminal window and I run SCT fMRI MoCo, I have the help here, and you can see that you can change the smoothness of each volume. You can change the number of iterations, the metric. In this case, as you can see, we use mean square, right? So I don't know if you remember, but I told you yesterday, well, if you're using images, never use mean square, right? So I'm doing the opposite here. And the reason I'm using mean square is because, in fact, the contrast across all the different fMRI volumes is the same, right? It's the same contrast. But then you could argue, well, you showed us that in some cases we see the CSF and sometimes we don't. That is correct. So maybe the mean square is not the best one. Maybe I should use cross correlation instead. So again, depending on your data, depending on if you're using, flow compensation pulse or not, you might want to play. You might want to adapt the metric that you're using for your motion correction. Right? Okay, so it took about a minute and a half. It's generating the quality control that we're going to look at together. Right, refresh, fMRI MoCo, and again, this is our before and after motion correction across the different time series. All right. So at this point, we can then finally segment the spinal cord on the motion corrected average time series. So this one outputs the mean fMRI MoCo, and that's the one we're going to use to segment the cord. And again, we're going to use the contrast agnostic method. So triple click, copy, paste. And for fun, we can see how this looks like on FSLI. So I'm going to open my fMRI MoCo mean just to see how it looks like. Just in intensity. Okay. So it's not too bad, but a segmentation algorithm might have hard time, okay, especially here, for example. It's very difficult to define what the contour of the spinal cord is. Same here. Same here. So, and these are not bad data, I've seen much worse. So, processing fMRI data is very tricky. Okay, so segmentation is done. Let's look at the QC. Go back to the index, refresh. And yeah, that's, reasonable. It's not perfect. You can see some slight leaking of the segmentation here. There as well. And maybe there slight over segmentation as well. So if you want, very, very precise segmentation, you can always redo your, refine your masks, but I would say this is a good start. Okay. Can also compute some metrics such as, oops, sorry about that. You can also compute metrics such as the temporal signal to noise ratio. So that would be fMRI compute TSNR, and you would do it on the fMRI time series and on the motion corrected time series. So let's run this command. And this command. And then we're going to display the temporal SNR results on the QC. So that would be the command while you run the fMRI TSNR. The second input is the MoCo TSNR. We're inputting the spinal cord segmentation just so that we can center each axial sub-figure on the spinal cord. And then we can refresh the quality control and then compute the SNR. So this is a feature that Sandrine worked on. Looks beautiful. And then you can also compute, you can toggle between so the number one here, it's tiny, but the number one, maybe like a note for next time is that it would be nice to have the actual file displayed on each overlay. If we see that this is fMRI and this one is fMRI MoCo, I think that would be quite useful. But so what you can appreciate here is that the temporal SNR is much higher in the motion corrected case compared to the non-motion corrected case. But I also don't want you to draw the wrong conclusion. Can someone tell me another reason also for a slightly higher TSNR in the motion corrected case compared to the non-motion corrected case? Because, as a starting point, the amount of motion was not terrible in this subject. So how come the TSNR is that different? Does someone have the answer to that? One? Anyone from the Neuropoly lab, hopefully? Abel, yes, I saw you moved. Please go ahead. You want to unmute yourself? Yeah. No? Okay.
Is there any smoothing involved, perhaps?
Sorry, who's talking?
Anugrah.
Anuga, yes. Can you please repeat?
Perhaps is there any smoothing involved or?
Great. Yes, you're right. And so where would that come from? Let's look at what we run. SCT fMRI MoCo, okay. But I showed you before that there is a lot of other parameters. So let's type -H to see what are the other parameters. And now can someone point me to the culprit here, to the parameter that will make your TSNR higher than what it appears or what it should theoretically be. That's a good signal processing quiz.
Can I answer?
Please.
I mean, here the default smoothing is 0, but I would say it's just the interpolation that is at the end.
It's this one, yeah. Exactly. So what you see here is that, what is a motion correction? Motion correction is you're trying to estimate, motion parameter for each slice, each volume, and then you're essentially correcting it. By correcting it, you're you're moving your image, and by moving your image, it means that you're resampling it, right? And there are different ways to resample an image. You can use nearest neighbor interpolation, you can use linear interpolation, spline interpolation, and so on. You can see that the default one is linear, okay? And linear interpolation, well, you can see that as, a small smoothing kernel, because you will take the signal of all the neighboring voxels and you will use that to interpolate your final matrix for the corrected volume. So to be fair, this is not a proper comparison. So, don't ever publish something like this because that would be wrong. Right? A correct way to assess motion correction in this case would be to rerun the motion correction, but using another interpolation. Let's try to do this actually. And yeah. Or actually, I want to call it differently. Can I call it differently? Do we have an O folder for this case? Yes, we do. O folder, and then I'm going to call it NN inter. It's gonna be interesting. It's an improvisation. I've never done it before. So the thing we're gonna do then is we are going to rerun the temporal SNR, but on the new fMRI MoCo, the one that is nearest neighbor interpolated. Maybe a note for next year that this is something we should actually talk about, because it's very important. It's a very important aspect. Maybe while this is running, I can just add it here. Computing, comparing TSNR on linear. So all right, so let's see now. So this is almost finished. It's a bit of time because there's a lot of GIFs to save. Okay, so now we can go to the NN interp folder. And I'm gonna rerun this thing here. Okay, and now I'm going to rerun that command But what I need to do is to specify that this fMRI TSNR will be in the parent folder, right? Okay, so I have a problem. Oh, yes, this one is also in the parent folder. So you see another example of an error. No such file access. fMRI mocomin. Indeed, because this one is in the parent folder. So I just have to add that dot slash to go to specify that this is in the parent directory, right? Okay, now we can open the QC, and this is going to be interesting now, because now we are comparing. Aha, you see I'm toggling before and after MoCo, and you see that the difference is like there is almost no difference, except for maybe the the slices at the very bottom of the scan. So you see, comparing these differences to these differences, it's only due to the type of interpolation in the end. It's crazy, right? So you see how easy it is to to draw wrong conclusions just because of a little flag related to the interpolation. Signal processing can be very scary sometimes. Okay. Any question? So if you do fMRI, then you're very likely interested in the spinal levels. And if you're interested in the spinal levels, then what you would usually do is, looking at your vertebral levels and then infer on, where the spinal levels would be, because there is, a good anatomical description of, where the spinal levels roughly are located compared to the vertebral levels. You can see that in many neuroanatomy books, from Gray's Anatomy and other types of books. So there have been, recent publications, notably the one from Frostel et al. That does some sort of meta-analysis of what is the correspondence between the vertebral levels and the spinal levels in human adults population. So we've recently updated SCT spinal levels to account for this description by Frostel et al. And if you warp the template on your fMRI time series, then you can have access to this like approximate location of the spinal levels. And this is what we're gonna do in the next slide. With the warp template, we're gonna set -A to 0 because -A corresponds to the white matter, and we don't care about the white matter here because we're doing fMRI. So it's just gonna be faster. And by running this command, you'll see what you're gonna get. Ah, I'm in the wrong. Okay, so can someone tell me what's the problem here? Another quiz. I know I'm annoying with my questions, huh?
You forgot to run something?
Not in this case. Good try though.
You're in the wrong folder.
I'm in the wrong folder, exactly. I need to go back to where I was, because you remember I did a little experiment. I created this folder. I went into this folder, but in this folder, if I do an ls, I don't have the files that I need in order to run warp template, right? Because I need my warp template to fMRI file, which is in the parent directory. So I need to go in the parent directory, and then I can run that. Aha, another problem. That's interesting. What is the issue here?
You did not run the registration command in the
Right,
last
thank
slide.
you. I love that. Love that. I did not run this, indeed. It's much better than ChatGPT. It's like a human aided image processing pipeline. Okay, so what we're doing here is the same as we did for MT and for diffusion MRI. We are registering the template to the fMRI scans. Okay. The input will be the T2S because we like to use the T2 star image from the PAM50 because we can see the gray matter. It's a bit closer to the contrast of the gradient echo EPI, right? So that's why we're using this one. The segmentation is the PAM50 chord, so that's the same. Destination is the averaged fMRI motion-corrected time series, and the segmentation is the one we computed. Now the parameters can be very tricky. You could spend hours and hours tweaking those parameters depending on your data. What I decided to put here for this course is the first step using the segmentation and the center mass. That's same as we did for the diffusion-weighted scans. Step 2, we rely on the segmentation again, B-spline scene, slice-wise, and 3 iterations. Then we have a third step that relies on the image, just because I don't trust the segmentation very, very much. We saw before that the segmentation was a little imprecise. So I want to give the image a little bit of credit to, do some like fine-tuning, fine adjustment of the registration based on the image. We're going to use the aggressive scene algorithm, cross-correlation matrix. We're going to give it 3 iterations, so that's pretty good. Gonna move quite a lot, and slice-wise. Okay, so it's finished. We can look at the results. Go back there, refresh. And so what we're seeing here is the averaged fMRI MoCo time series, and then I'm toggling now with the PAM50 T2 star, right? So again, I like to put my cursor on the boundary of the spinal cord to inspect. So did a pretty good job. So you see that it's a very tricky one because we see the CSF in some regions, we don't see it in some other regions. So makes it very difficult for a registration algorithm to deal with that. Very difficult. Nevertheless, here it looks pretty good. Here you see I'm not so happy about that. There seems to be some, overestimation of the spinal cord boundary. Here, I don't know if you see, it's very subtle. You can try that you can maybe see that on your screen. But the gray matter is actually a bit deformed. The gray matter is kind of moved towards the, the top part of the cord. So it's not perfect, but again, you're dealing with, very difficult types of images. You might smooth anyway afterwards, so. But that's what we get so far. I would say this is something we can work with. Okay, so now we can go back to this warp template, run it, and hopefully I won't get any error. Right? We are opening the QC. So that's our white matter registered to the fMRI, to the averaged fMRI MoCo. Looks reasonable again. And the thing I wanted to show you is the actual vertebral and spinal levels. So let's open them. I'm going to open Facialize on my fMRI Moco Min. Okay. And then I'm going to open two files. One file will be the vertebral levels, which is this one. The other one will be the spinal levels, which is this one. Okay, we're gonna give it two different colors. One will be green, and the other one will be blue. Okay, so what you're seeing here in green are the spinal levels. So that would correspond to spinal level C4, spinal C5. And then if I display the vertebral level, you see that they are slightly shifted, right? Because the vertebral levels is based on the location of the discs, right? And so that point, for example, corresponds to vertebral C3. Okay. Does that make sense? And then you can use that in your analysis. You can use the spinal levels if you want to do, you know, group level analysis and group level registration based on the spinal levels, as opposed to the vertebral levels. Any question?
May I ask a question about the group level analysis?
Yes,
Like,
absolutely.
how do I proceed after like yeah from the SCT warp template? So should I use the SCT apply transformation from the the motion corrected image of yeah, using the template that we got from the PAM 50 T2 transformation? What do you think, like for the group level analysis?
Yeah, so this is a good question. So first, so the first thing to consider is that, and this is something we're going to talk a little bit after, is that what we're showing here, and what we, you know, when I say this is the spinal levels, these are, you know, the probabilistic location of the spinal levels based on the correspondence between the vertebral and the spinal levels from the Frostel et al. Paper. Okay, which is not the same as saying that this is the spinal level of your subject. In order to know the spinal levels of your subject, you would need to identify the nerve rootlets, and it's much harder to do, but that's something we're going to touch base on later in this course today. Okay. So that being said, given that there is a one-to-one correspondence between the vertebral and the spinal level, in fact, you could do a group level analysis by, you know, merging your first level maps in the PAM 50 template. Okay. And then in the PAM 50 template, you can simply display the spinal levels of the PAM 50. That would be equivalent of, you know, an equivalent of doing a group level analysis. And again, you can do that because the one-to-one correspondence will be the same across all your subjects, because you're basing yourself, essentially the spinal levels are purely based on the vertebral levels. That, you know, does that make sense?
Yeah, okay.
Yeah. No, it's a bit complicated, but just to answer your question properly, what you're gonna do next is, you know, we can open the PAM50 template, or let's say we can apply. Okay, so let's say that you have your first level analysis, your first level results, which could be let's say that fMRI TSNR is, you know, is an output, or like this one. fMRI MoCo TSNR. Let's say that you want to put this one in the PAM50 space. Okay. We're going to apply transfer, as you said, SCT apply transfer. What would be my input? Hello?
The input will be the template PAM.
No.
No?
No, because in this case, what you want is to bring your subject's first level analysis onto the template to do group analysis like that, right?
Right.
So your input would be, like in this case, I'm just, you know, choosing a dummy example. I'm just taking the TSNR data, but in your case, you could take first level analysis, for example, or like, you know, your T-score maps, or, you know, things like this, right?
Right.
And the destination here will be the PAM50 space, right?
Right.
So in this case, this is our Spinal cord toolbox, data, PAM50 template, template, PAM50, star, for example, and the warping field. Which one will it be? Can open my Finder window. I'll go to the fMRI. You see that I have something called warp template to fMRI. Is that the one I should take?
Or not the fMRI to template?
Exactly. That's exactly it, right? Copying, pasting, and yeah, enter. Okay, and now we can view. So what we're going to open is the PAM50 T2 space. Sorry, T2 star. And then the registered fMRI MoCo TSNR, which is now in the space of the PAM50, right? For that particular subject.
Right.
And you can imagine that you can do that for all your subjects and then average together, right?
Right. I see. Yeah.
So it's a bit ugly because, you know, the resolution is horrible. But, you know, if you want to do group study, that hopefully will, you know, be a bit more meaningful. Does that make sense?
Yes. Thank you.
Any other questions?
Can you please explain the motion correction? I didn't get how you change the parameters or the algorithm you use, because in the context of a patient with compression, I think the motion correction before we create that mask is important. So could you just explain it again, why the default was not appropriate and then you had to change? I was a little confused.
Are you talking about the interpolation?
Yes.
Okay, so the interpolation has nothing to do with the actual motion correction algorithm or like quality of correction. It has to do with how you're going to resample each of the corrected volume into like, you know, matricial space, into a discrete space. And it has to do with, you know, again, partial volume. That's another example of partial volume. It's, you know, when we compute the temporal SNR on, or maybe I can, okay, what I can do is open an fMRI time series. Maybe I already have it. Okay. fMRI Moco. So I'm opening this one, which was interpolated linearly, okay? And then this one, which was interpolated using nearest neighbor, okay? So again, this is the exact same parameters for the registration, because motion correction is essentially a registration algorithm. The only thing that changed is at the end, you know, how you're resampling your image. So let's put this as at 1000, the other one the same. Okay. Going to zoom in a little bit. And then I'm just gonna toggle. Yeah, we don't see. Oh, okay. So that's a tricky question now. I have another quiz question. I'm toggling on and off between those two images, and I don't see any difference. Can someone explain me why? Even in the last slides. As are they not aligned? Well, looks like they are aligned, but there's also something that has to do with the way the algorithm works. So the way motion correction works is that you'll take, you know, you have like, let's say 50 volumes. You'll take volume 2, and then you'll align it to volume 1, volume 3 to volume 1, volume 4 to volume 1, and so on.
Yeah.
Right. So there is some reinterpolation going on if the motion parameters are not 0. But that interpolation is starting at volume number 2, not at volume number 1, because volume number 1 is the reference volume. So what we are doing here is that we are comparing two types of interpolation on the first volume. They are essentially the same, right? But now if I go to the second volume, I might start seeing some differences, and indeed I do. So I'm just going to zoom in a little bit. And if I toggle those two on and off, you might see some very subtle difference between the two volumes.
Yes.
Do you see that?
Yeah.
And what you're seeing here is just the effect of the linear interpolation. Again, linear interpolation is, you know, if you look at this voxel, it will receive contributions from the neighboring voxels here. So it's like a very small smoothing kernel, right? That a bit more clear?
Yes, I will give it a try on our data and see what it looks like.
Yeah.
Thank you.
These are, you know, these are fairly complex signal processing considerations. So it's, you know, it takes time to digest all this information, and you know, by time I don't mean hours or days, but more like months or years. So, you know, being familiar with all these concepts is really a matter of years of experience with you know, neuroimaging processing.
I have a question for you, Julien. Would you recommend basing the motion correction on the 0, the first volume?
That's a great question as well. I know that some groups like to base the reference image to the average volume of the time series, for example. There are different arguments. The first argument is maybe the first volume will be crap maybe there's been like horrible artifact, and then you're basically relying on the relying on a crappy image for your entire motion correction, which is horrible. It's like a disaster. So again, look at your data. You know, if you consider that the first volume is a good one, then maybe that's something you could do. Another consideration is that let's say you acquire a field map because you want to correct for susceptibility distortions. Well, is your field map acquired before your fMRI time series or after? If you're running a resting state scan that lasts for like 15 minutes, it's very likely that your subject will go from there to there in the scanner. A very big motion. Okay, so if your field map is acquired right before your fMRI scan, then you'd rather rely on the first volume of your fMRI scan for the fMRI motion correction. But if your field map is acquired in the end, then maybe you want to rely more on the last volumes, right? So, these are two examples of things to consider, but there are more, but since you're asking the question, I'm just going to show you that with the motion correction, type SCT fMRI MOCO, there is a flag that where you can specify the target number that you want to register it to, right? So by default, we register it to the first volume. If you want to register it to the volume 300, then you can also do that. Another approach, which is one by default, is that there's an iterative averaging, is that the target volume becomes, it changes all the time. The first iteration is that volume 2 is registered to volume 1. But then after this is done, volume 3 is registered to the average of volume 1 and volume 2 registered to volume 1. And so on. Right? What's the advantage? The advantage is that you have a target that is, less noisy. That's essentially it.
Can I ask a follow-up question to that?
Yeah, of course.
What would you be your strategy if you had like a large group of patients that you had to do the same analysis on, and would you do the B0 mean, or do you want to do both and see what is best, and maybe take like a couple of samples and look at them, or how would you address?
First thing, I would look at my data. That's always my answer. Look at your data, depending on the amount of motion, the population, the amount of noise, the amount of artifacts, the coverage, the size of the field of view, number of slices, resolution. All these things matter. So there's I would look at my data, I would try a couple of algorithms, I would compare my QC, and then I would make a decision based on that. Okay, any other questions? So it's 11:52 here in Montreal. I think it's a good time to maybe make a break, and then we'll reconvene for the for the final sections of the course. I think we have we have time, so let's make a 45 minutes break, and then we'll reconvene at let's say 12:40. Let's do 12:40. 12:40 in Montreal, obviously. Yes, thank you, Jan. All right, take care, everyone, and see you soon. Zoom recording. Good. All right, we're back. Any question, anyone, before we resume our course? Okay, so other features. Quality control, yes. So we've been using that thing a lot. So you should be familiar with the quality control so far. I just want to add that there's a lot of different like functions that have their associated QC that we have not necessarily covered today, like SCT image stitch or like straight and spinal cord. So I invite you to look at them, and they could be useful. One nice thing that is mentioned in the speaker note here is that let's say you process 200 subjects, and then your collaborator is like, oh, how does your processing look like? Is it were your segmentations good, bad? And another scenario is that, let's say you want to publish a paper, and then reviewers are asking you about the quality of your processing, or even if the reviewers don't ask you that, you might want to share the result of your processing without necessarily showing like some faces for like ethical reasons. Then sharing the QC reports can be done very easily. Going back to your to the folder where your QC report is, it's essentially an HTML file. If we click on it, it will open the QC report. It's an HTML file, and then there's a bunch of dependencies and the pictures that are displayed in the QC report. What you can do if you want to share your QC report, you just take this folder and you can zip it. And then that's something I very often do. I take my my zip file. So this one is pretty large, 22 megabytes. But you can, if it's smaller, you can email it. You can upload it on a GitHub issue. You can put it on Dropbox or Google Drive, and then you can share this thing pretty easily. And it's a self-sustained object. So if you send it to a collaborator, they can unzip it, open it, and then look at the QC. It makes it very transparent and easy to share the results of all your processing. Another thing also is that there are some features that we have not discussed yet, but that we are going to discuss now, which is about assessing the quality of the processing. So if I go back to my quality control image, you see that there is a column here called QC, and there is some features here that says pass or fail or artifacts, and the key to activate those is F. So if you click on F, then it will create a little emoji for pass, and if you press F again, it will change to a fail, and if you press again, it will change to an artifact. And if you press again, it just goes back to the original. And so let's say if you want to quickly review some segmentations, then you can go there, pass, 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 pass. And let's say this is fail. So you just go fail. Let's say you're not happy with that. Let's say fail, pass, and so on, right? So you do that for all your subjects. And you can also do this for your deep seg function only, right? If you only want to do that for deep seg, then you can go to search and you type deep seg like that, right? And it will only show the thing with deep seg, right? And then you can then easily QC everything that is deep seg. Or if you want to only QC the T2 images, then you can just write T2.nii. And then only the T2 are, showing up here. Right? And once you do your QC, you can then save all the things that failed. Click on Save Fails. It's on the desktop. And then on the desktop, you have a YML file that you can open. I'm going to open it with a text editor. And then it shows all the files that you checked as failed. And this is very useful because imagine that you have 500 subjects to QC. You take an hour, QC everyone, and then you check the one that are failed. And then you can use that YML file to launch a process that will open up an image viewer for each of these subjects sequentially. And then you can correct, it will open up the segmentation, and then you can manually correct the segmentation, save it, and then go to the next subject that is listed here. So it's a very nice way to help you, manually correct some of the failed processing. If you want to know a bit more about this workflow, we're going to talk about this a bit later when we talk about analysis pipelines with SCT. Next slide, I wanted to... Oh yeah, there's a question from Merve. How can I remove some subjects data from QC in an efficient way? Sometimes I run things over and over and QC gets very busy, and just removing that subjects folder is not enough. Right. So there are different ways to do that. The way we like to do it in our lab, I can show you an example of the Spine Generic project. The Spine Generic project has an analysis pipeline and also a data set associated with this. If I go to the data set, multi-subject. So these are like 267 something subjects from different sites. And some of these images we had to exclude because of artifacts, right? But the last thing you want to do is manually remove a folder. Never do that, right? Because it basically, let's say in 2 years from now, then maybe you'll find an algorithm that can get rid of this artifact, right? Or maybe you want to train an algorithm that detects artifacts. So in other words, never manually get rid of your images. What you want to do instead is to create a file that will encode the files to exclude and why you're excluding them. So here, for example, you see there is an exclude.yml file. That file, if you open it, has, for specific category, for example, MTR, we excluded the Stanford data because they use a different TR, or we excluded this particular subject because the MR technologist used a different coil. That was a mistake. So, you'll always have some issues with your data, some mistakes from the MR tech, some artifacts, whatever. So the key is just to document this properly. And then once you document it, this YML file here, you can use it in your analysis script, right? Your batch script will loop across all your subjects, and you can then have an algorithm that says, remove, like ignore all the files that are listed in this exclude.yml file, right? So you can do that very easily programmatically, and this is the most elegant way and most reproducible and transparent way as well.
Maybe on this, how I understood the question was in the same QC report. If you rerun the same analysis over and over, your QC will get kind of overwhelmed with different analysis. Was there a way to remove those subjects in the QC report directly?
Yes, exactly. That was my question. Thank you,
yes,
Santi.
sorry.
Thank you. This is a very good trick too. Yeah.
Okay, sorry, I misunderstood. Well, I mean, one way is to then create a new QC for your rerun. You know, QC number 2, or like QC with a date or whatever. I mean, yeah, that it depends on how you organize, like what kind of things you want to test and so on. Yeah, we can
see.
discuss that
Okay,
further.
thank you.
Pleasure. Okay, moving on. Another feature is the Facialize SCT integration. So let's look at it. The Facialize has a way to use scripts or plugins. And so we created one with SCT that is compatible with Facialize. There's a YouTube video that you can watch that explains how to do it, but I can also show you live how that works. Let's open Facialize. Okay, so that's Facialize window. We can also open an image. Why not? Let's open a, I don't know, like this DMRI. Yeah, let's open this image. Okay. And then we're gonna go to where is it? Forgot where it is. Oh yeah, File, Run Script. And then you go to Spinal Toolbox, installation folder, contrib, FSL integration, and then SCT Facialize scripts. And you can open it. And then your Facialize is transformed. There is like a new panel that shows SCT with a different set of functions, including PropSeg or SCT DeepSeg SC. And the way you use it is, if you want to select the file that is highlighted, you just need to click on input file, select the contrast, EWI, output folder. Well, fMRI is not a great output folder, so I'm just going to select a more appropriate output folder for this, which is data DMRI. This one is good. Run. So it runs SCT DeepSeg, and then that's the result of the segmentation. And then you see that the file appears at least here. And then you can do that, with like other functions and so on. We might add more functions on this plugin if you like it, which is why we asked you to fill up a poll to see, because if nobody uses that, then we're not going to spend time implementing features in there. But if you like it, and you tell us that you like it, then we might dedicate some time to improve those features. Okay, another feature that that is relevant in some cases is smoothing along the spinal cord. For example, if you do fMRI, it's quite common to smooth along the superior inferior direction to increase the effect size of fMRI, like T-score maps, for example. But the problem of doing superior inferior direction smoothing is that because the cord is curved, you'll end up with a different smoothing quality depending on if the cord is orthogonal to the axial plane or curved. So ideally, what you want to do is smooth along the spinal cord, right? That's a more natural way to smooth. Very similar to the surface-based smoothing in FreeSurfer. So that's possible with the SCT Smooth spinal cord. And we're not going to do that here because of the interest of time, but you can run the segmentation at the first pass, and then use that segmentation to run the smooth spinal cord command. And then you can rerun the segmentation or any other type of analysis now using the smooth image, right? Another, yes.
Sorry, if I have like a pipeline that I'm writing, can I put in that section of smoothing, and it will not alter like a good-looking spinal cord. It will only alter the ones that are, you know, faulted, or will it affect good-looking spinal cord in another way?
So I guess, you know, the questions has multiple meanings to it. First of all, what do we mean by affect? You know, like smoothing does affect every image, you know, the good-looking ones and the bad-looking ones. And it affects it by, you know, effectively averaging across multiple voxels. In some cases, you don't want to do that. So if the rationale for smoothing is, you know, strong enough that you would like to apply some smoothing, then the other part of your question is, you know, how is it going to affect the good-looking images? Well, then we can question what is a good-looking image and what are the features that you want to preserve when you run some smoothing, right? There are different motivations for smoothing an image. One motivation is to improve the precision or the robustness of automatic segmentation, right? Automatic segmentation of the spinal cord or the gray matter, right? If you have a very, you know, noisy gray matter appearance, then maybe smoothing will help. But then you need to realize that by smoothing, you're also, you know, altering a little bit the actual shape of the gray matter or the spinal cord along the superior and inferior direction. If you want to smooth because your goal is to, I don't know, extract some metrics along the spinal cord and then, you know, extract them in a more reliable way, then, you know, that shouldn't be a matter if you do that on, you know, in like, you know, good looking or bad looking images. But we could also argue that, you know, why not doing this averaging after the fact, right? You run the extract metrics and then you apply some statistics, some regularization, and so on. So, you know, it's not entirely clear to me where you're coming from, you know, what's your rationale for smoothing, but, you know, all I can say is that it really depends on, you know, why you want to do that and what are the, you know, questions that are addressed. Point from Ajay, another option may be denoising. Yeah, indeed, denoising. I mean, that's essentially what we are trying to do here in this example, you know, to improve the quality of the segmentation. If it's too much affected by noise, then that could potentially help. Yeah, yeah. Another feature that, you know, I have to say I rarely use, but you know, if you want to show a nice figure for your article and then your subject you know, is not perfectly aligned because of a, you know, slight curvature of the cord in the right-left direction, you can use an algorithm called SCT flattened sagittal. And what it will do is it will basically take the, you know, the input segmentation and then align it align it along the right-left direction so that you have, you know, your cord is visible in the medial plane all along. And then there are also other operations that you can do that we did not cover in this course. You know, mathematical operations, root mean square, some Otsu algorithm for thresholding, and some erosion, dilation of binary masks, and so on. You can also manipulate your images. You can split along different dimensions. You can concatenate along specific dimensions. You can stitch your images as well. If you acquire, for example, a large field of view for the cervical and then the thoracic, and you want to stitch those two parts together into a single image, that's what stitch would be used for. Header, you can look at your header, you can copy headers, copy S form or Q form, and so on. That's the stitching algorithm that I just told you about. That was implemented by collaborators at Technische Universität in Munich. Then, we'll encourage you to just see what are all the different functions, typing SCT tab, and then it lists all the SCT functions that are there. So it takes a bit of time to basically read all that. But these are also listed on the Spinal Cord Toolbox website. If you go on the Spinal Cord Toolbox, then you go to command line tools. They're all listed there and categorized as segmentation, labeling, and then you can click on one and get all the different documentation. So it's another way to list all the functions that are available. If you're like a tech savvy and you want to develop some APIs or use some of the SCT's APIs, then you can also activate Python's the Python that we used that comes installed with SCT. And that way you run the unit tests, for example, or you can also essentially develop within the SCT environment. We have a question from Barbara. Does the stitching require images of the same dimensions? It does not. You can actually stitch an image, that is 200 by 200 by 300, and another that is, 100 by 100 by 400. As long as they all lie in the same absolute or like physical coordinate space, then you can stitch them, because the stitching relies on the Q form of the nifty header. All right, any other questions before we move on to the next section? Analysis pipelines with SCT. That's my favorite section here. That's my favorite section because this is about, scaling up your analysis from one subject to, hundreds or thousands or even more, subjects. And it's really a way to make your entire processing reproducible, transparent. Let's say you run your analysis, and then 6 months later, a reviewer asks you to to change one parameter, or like to check like what if the results hold if you use a smoothing of 5 millimeter kernel compared to 4 millimeter kernel. And then instead of going through all the each of the 200 subjects and then rerunning manually all the commands that we did today, you can just change one line of code and press enter, and then it reruns everything, and it recreates all the QC report, all the figures and everything. So that's really the way science should be done. And we're gonna look at that together, how you can reach that stage of analysis. The first requirement is to organize your data, to make sure that they are organized in a standardized way, standardized convention. One convention that we adopted in the lab and in SCT recommendations is BIDS, which stands for Brain Imaging Data Structure. And it's essentially a way to name your files. For example, you have a README that describes your data set, some TSV, and then each subject is organized under a folder. And then inside a folder, you have the ANAT images. And inside ANAT, you have, let's say your T1, your T2 star, and so on. You might also have DWI or fMRI and so on. Okay. So let's see how we can run the processing using BID's organized dataset. So first, we're going to change our folder now, and we're going to go to the multi-subject folder. So I'm going to go to that dot, and then if I tap multi-subject, here it is. LS. Okay, you see that my data are under data. I'm going to set the proper permission so that I can execute the shell script properly. So copy, paste it, okay. And then I want to see what's inside my folder data. So I'm going to write tree data. And then it shows that my data consists of 3 subjects, sub 01, sub 03, sub 05. And inside each subject, I have a T2, an empty off, an empty on. And it's the same for all the subjects, okay? So you see where I'm going here. We're gonna create a script that will crawl within that directory and loops across all the subjects and then do some processing on everything that's called underscore T2 and everything that's called underscore ACK empty off and ACK empty on, right? Because that's the same across all the subjects, right? So let's see how our process data script looks like. We can open it using that command, or you can also open it from your Windows Explorer. So I'm going to SCT course data, multi-subject, process data. Going to open it with sublime text, for example. Okay, that's how it looks like. So it's a bit overwhelming at first, but I'm not going to describe all the functions, but there are functions inside my shell script that I'm going to call later. This one is called label if does not exist. What it does is that if there is a manual, like a manual correction of a segmentation, then it will copy it where it should be. If none is found, which is pretty much what you start with when you start off, you know, from a you know, like a pristine data to process, then it will run vertebral labeling, and then it will create some like 2 point-wise labels at vertebral level 3 and 5. There's another function that works in a similar way. It will look for a manual segmentation. If there is none, if there is one, then it will copy it. And if there is none, then it will run SCT deepseg to create the segmentation, okay? So these are the functions, and the script actually starts here, okay? So what the script does is that it will retrieve the subject name, sub 01 or sub 03, and so on. It will display some useful stuff. And actually, you know what? I'm going to run the script because it takes a couple of minutes. So I'm going to run it, and while it runs, I can explain this to you. So copy, paste, and that's it. So it's running. Okay, let's go back to the script so I can explain that to you. The first part is the T2 weighted, okay? And we'll go to the subject that has been identified. So subject 1, 2, 3, 4, 5, and so on. It goes to the ANAT directory. And then it will create a variable called file T2 that is composed of the subject name and T2, T2W. And you see that image is going to be, for example, that one, right? Sub 01 underscore T2. That's it. That's my variable file T2. What do I do with this variable? Well, I'm inputting it inside this shell function that I described above, right? Segment if does not exist. So since this is a pristine data set, what it will do is that it will run ST-DeepSeg on this file. Right? And then it will create another variable that has the name of the segmentation on the T2-weighted image. It will create some labels using another shell function that is defined above, okay? And then it will register to the PAM50 template. So this is a function that you know now because we saw it during the course, but you see that I'm not going to type it in the terminal. The script is going to run it for me automatically, okay? And what we're going to input in this is the T2-weighted image, the segmentation that we just did, the labels that we just did before, some parameters, and then we want the QC output. And then at this point, we can warp the template, and then we can run some processing. For example, compute cross-sectional area averaged at vertebral levels 2 to 3, and then call it CSA.CSV. Okay, and we're going to append it so that every new subject that is processed is going to end up in this single CSA file. Any questions so far? I know this is a lot of information. Okay, so I'll continue with the description of the script. The next part is called empty, because we also have empty data that we want to analyze. So we're going to create variables the same way we did with the T2-weighted image. We're going to segment the empty one. We are going to create a mask. So all that we already did. I already showed that to you in the course yesterday. We're going to crop the data so that processing is faster. Then we're going to register the MT0 on the MT1. And then we're going to register the PAM50 template to the MT1 data. And for that, we're going to use the initialization warping field that we just calculated above in this section. Is that clear for everyone so far? After we're there, we can rename the warping fields so that they have, a name that is a bit more pleasing to the eyes. Template to MT, and then MT to template. Right? Then we're gonna warp the templates labels, and we're going to compute MTR, same as what we did before, and we're going to extract MTR in the dorsal column, averaged between vertebral levels C2 to C5, using the maximum a posteriori method, and we're going to output it into this CSV file. Again, we're going to append it so that every new subject that is processed is going to end up in the same CSV file. This is going to be much easier for you. Imagine that you have 1,000 subjects. It's much easier to deal with a single Excel sheet than 1,000 Excel sheets, right? And that's it. At the end is just some verification code, some display if all the files have been properly created. If not, it creates an error log and so on, right? So I'm going to close this bash script, go back to my terminal, and you see that it's still ongoing, right? Processing is still ongoing. So while this is ongoing, I can explain to you a little bit of the things that we're seeing. Okay. So that was our syntax. I wrote something called jobs 3. What does that mean? It means that we want to parallelize the process across our CPU cores. Nowadays, when you buy a computer, it usually has multiple core CPU, and each core can run a particular process. And so let's say if you have 20 core CPU, then you can run 20 processes in parallel, right? Now there is a sweet spot because the RAM, the amount of memory also plays a role. So you don't want to saturate your RAM. So there is a sweet spot depending on the process, the amount of RAMs that you're using, the size of your images, and so on. But you can get more information about your system below. If you run the same command that I did, you will get to different values because you have a different computer than me, right? It will show the amount of CPU cores that are available. It will show you the total RAM, the one that is available. And then it will show the different input arguments of the functions. So you see that I'm using 3 cores out of 12. So I should be fine, okay? And then this I'm gonna talk about it later. And then it runs the processing. So you see it processing three subjects in total, including 3 in parallel. So this is ongoing. And while this is ongoing, I can show you what are the files that are being created. So, or maybe I can pause a little bit and take some questions, or like look at the chat. Let's see. Any question so far?
Yes, I do have a question. On your batch run, it looks like you have it set up for the structural analysis and the MT.
Yeah.
Do you have like a batch processing that include the functional analysis based on what you just showed earlier today?
So
especially the first part of the motion correction and the spinal cord segmentation for the epi scans.
right. So I'm going to give you a very unsatisfactory answer again.
Okay.
I don't want to do that because if I do that, you will use it. And if you will use it, it will not work with your data because every data set is different. Every analysis script is different and should be different. So, I'll be nice and I'll show you like how you can, at least change it. For example, let's open it. Let's create an fMRI section on the fly, right? fMRI. Well, you will want to create, something like this. Oops. File fMRI. You'll have your subject name and then underscore probably something called fMRI. Is that right? That's probably not the beads. So if you don't know how the beads fMRI is called, thing to do is to go to the beads.
Yeah,
right?
that I can do, depending on the run, right?
Application, yeah. First line, fMRI. There it is. That's how it should be called. Underscore task. Okay, so underscore task. I don't know, like fist clenching underscore bolt. Right. And then you want to run some motion, or like first before the motion correction, you want to bring the T2 segmentation in the space of the fMRI scan, right? And that you already have from the course material, right? Because that's something that we saw earlier. And it corresponds to this line here. Or like first you want to average the fMRI scan, right? So you basically can copy this line, bring it here, replace that with something like, you know, I can do this. File fMRI, but NI GZ in T, and then it can be called underscore min, right? So I forgot something very important here. Can someone tell me what I forgot? It's a bit of a tricky question. But if I launch my script, it's gonna crash.
Can I answer?
Yes.
Yeah, changing the directory.
Exactly. Because with BIDS, your fMRI scans, I don't know if it shows it here. Yeah, you see, with BIDS, the fMRI scans are under the func directory. So what you want to do first is change to func directory. cd /func. That's it, right? And then this is going to work. So you see that you can build the fMRI section yourself by editing this, example template. And you can put here whatever you need to put to run your fMRI analysis based on, the examples that we saw before. But again, this is only an example, and it should absolutely be adapted to the data set that you're processing, if that makes sense.
Yes, thank you. Yeah.
Sure.
Excuse me, can I just ask where you put the file name of the subject? Are you supposed to have the .nii.gz there? Doesn't it duplicate it when you call file fMRI?
That's a good question. So that's a good question. If you look at how we define this, how we define those variables, is that we omit the .nii.gz, right? We omit it. We only put the base name. So we have to add it afterwards, right?
It just meant lower down when you define file fMRI, you have the .ni.
Yeah, that's a very good point. That was a mistake.
Okay, thank you.
Exactly. Good catch. All right. Any other questions? Aha, you see, it finished. So it took a bit, it took about 11 minutes. And now is the fun part because we can look at the results. But before looking at the QC report, I just want to show you a little bit where the data are output. So the command that we run was script process data and then config.yml. So let's look at the config.yml file. Let's see what's inside. We can edit it, or we can also use cat config.yml, which is a Unix command to show what's inside the file. Well, to be honest, it's not very user-friendly. So I'm just going to open it with a text editor. It's going to be a bit easier. So config.yml, let's open it. And you see that there are different definitions. There is path data. This is where the data are located. Path outputs. This is where you want to put the output. If you have manual corrections, they are going to end up there. And then some prefix jobs for. You see that we overwrote these variables because we wrote jobs equals 3. So that one was overwritten, right? And you can read more details here to how you can adapt your config YML file. So I'm not going to save it. Let's look at our output results. Output folder. You see this one was created. You have a bunch of stuff in there. One thing you have is the data processed. So this is where all the data was copied and then processed in there. So all your physical outputs are located in this folder. So that could end up being a very, very large folder of multiple gigabytes or sometimes terabytes if you have a lot of subjects and a lot of images, okay? That's the log folder that shows you a log file as the data are being processed, so you can monitor it for each of your subject and for the general log. So the general log is a copy of what you see on the terminal, okay? And each individual log is the output of the processing for each particular subject, okay? So in this case, we're talking subject 1, and it shows you all the commands that were run for subject 1 with the outputs. And if you have some crash, some issues, then this log file is becoming very useful for it, right? Another folder is the QC report, which we are going to look at now, or maybe to look at in a couple of. Yeah, let's look at it now actually. So index. Okay, so this is the QC report for the batch processing script. Okay, and you see that we have all the subjects that are in like random order because this was it's a chronological order, but you can sort it by subject if you like. Or you can also sort it by functions. Or you can also, and the nice thing by sorting it by function is that then you can QC very quickly all the segmentations. So here we are looking at the segmentation of the chord on the T2 for subject 1. Looks good. On the MT1. And then for subject 3. On the MT1. Subject 5, sorry. On the MT1, and so on. So the way I usually do it is by looking at the same types of scans. It makes the QC easier. So let's say, let's just filter by T2 weighted. And then if you have like 200 subjects, then you would basically go through subject 1, subject 2, subject 3, and so on. Right? Makes it easier. And then you can have your left finger on F, so you can check pass, or like fail if you like, and then pass, and so on, right? And then that's how you QC your different processing. Same for the vertebral labeling, pass, pass, pass. Actually, no, fails here, right? There's a problem. So fail. And then register to templates. Looks good. So pass, 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 or pass, 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 and so on. Okay. So we have one fail, and then you can save the fails, and then we're gonna see how you can address that later on. Any questions so far? Okay. So we did our QC, and obviously what you want to look at is the results. So if you go to output and now under the result folder, you're going to have two files. One is the CSA, which corresponds to the cross-sectional area for each subject, because you remember we appended all the subjects on the same file. So you have subject 5. The mean cross-sectional area is 68. Subject 1, 82. a bigger spinal cord. Subject 3, 61, smaller spinal cord. All right? And you have the same for MTR in the white matter, in the dorsal column. That's the other file here. And here, these are, each line corresponds to a subject. And you can see that the MTR values are much more stable, which makes more sense because we know that people have, different sizes of spinal cords, but the white matter microstructure is fairly stable across, humans in the dorsal column. So which is why the MTR is relatively stable across our 3 subjects, right? But then if you have subjects with pathologies, with lesions, degeneration, then you would start seeing this MTR with abnormal values. Sounds good? So I showed you how to run the QC report, and then how to add a cross if you're not happy with the segmentation or with the labeling. Okay. Now we're gonna see what we do with this. We're gonna click save fails. So I'm going to reopen my QC report here. I'm going to click on save fails. Okay. Actually, I already had one before, so I'm going to delete this one. This. Okay. And then we can use a script called manual correction, with which we will loop across all the subjects that were marked as failed segmentation. And you can easily download it using this command. So SCT download data -d manual correction. So let's do this. Done. And then ls manual correction. You can see that now there is a folder within which there is a couple of files in there. And we are going to run. So we can run the help of this script to see what is available. You see that there is a lot of options. So I'm not going to describe all these options, but I'm going to show you how we can use that script with the analysis that we did together today. So I'm going to the next slide. And then I'm going to run that command here, which runs the manual correction script. Okay. It's going to use the qcfail.yml. So this is the file that we downloaded, right? It's located on my desktop. So I need to point to the right location. Path images is the location of where the data have been processed. And path label is the location of it's the same location, because this is where we're going to actually that's not very intuitive. Maybe Jan, we can probably make a note about this thing here. And then path out is the location where we're going to output the manually corrected labels. So I'm gonna run that command. I need to enter my name because I'm the person who will modify the segmentations, and you want to keep track of that. Let's say that you have a grad student who is doing the manual corrections, and then 2 years from now you want to revisit your data set, you want to revisit your analysis, and you want to know which segmentations have been modified and by who, right? So it's very important to keep provenance of that. Julian, run that, enter. And then it tells me stuff. In Facialize, click on edit mode, correct the segmentation, and then save it with the same name. Okay. So it's opening Facialize. And this is, I think I did not point to the right config YML. I think that's what I did. Yeah, that's fine. Okay, so you have a segmentation here that you want to correct. So you're going to click on edit mode. You can go to this thing. You can zoom in if you want. I like to look at the axial plane when I correct the segmentation, making it a bit smaller. And then you can add a pixel here. Then you can change slices. You can add pixel there. I'm just doing it, you know, coarsely just for the example. Okay, and once you're done, you're gonna save it. Click on save. You want to overwrite, and then you can quit. Then it says that the sidecar has been updated, and so on. And then it generates a QC report. And that QC report will show all the manual segmentation that you did on the problematic subject. So here I only corrected one subject, but if you need to correct 20 subjects, it would show the 20 subjects here. You remember that I put my name somewhere, and so where is my name encoded? Well, it's encoded in the data processed. Sorry, that would be output data processed.
Maybe under data, if you go 2 folders up, yeah, data, and there is supposed to be derivatives, yeah.
So you see that the BIDS convention is to create, is to use a folder called derivatives that mimics the organization of your data inside a subfolder. So in this case, you want to call it labels, or you can call it label spinal cord, you can call it whatever you want. Inside labels, I have subject 5 because this is the one that I manually corrected, ANAT. And then we have the segmented spinal cord. Okay, that's the one that I corrected. And you have a JSON file that says generated by Julian Cohen-Haddad with the date. Okay, so a note, Jan or Mathieu, this is wrong because we should also display the actual, the original segmentation in the JSON file, right? So that's a mistake. Because if you go to sub 05 ANAT, oh no, so it's not data, so it's processed. Output data processed sub 05 ANAT, and then the T2seg JSON. You see that it says generated by Spinal Cord Toolbox. So what we want is to append the manual correction to this actual field. So that's a mistake in our current workflow that we're going to correct. But for now, at least you have, provenance of who corrected the manual segmentation for this particular subject. Okay. So the question you have now is, what do you do with this? Well, what you can do with this is rerun your processing. So you're going to rerun the SCT run batch exactly the same way you did it before. So triple click, copy, paste. So you see I have the exact same information, processing 3 subjects in total, blah, blah, blah. So all looks the same, except that for one of the subjects, subject 5, it will use the manual correction, the manual segmentation that is present in the derivative folder, as opposed to rerunning it automatically, right? And you can verify that this is the case. You can go to output correction, which is the new output folder for my processing. This is the first one we run, and that's the one that I'm currently running. If you go to the log files, these log files are being created on the fly. There is one for subject 1, subject 3, subject 5. Okay, let's look at the one for subject 5. The first process to run is the spinal cord segmentation. And you can see that it's looking for the manual segmentation and it found one. And so it's going to use it instead of rerunning the segmentation for that particular subject. Whereas if I pick another subject, subject 1, I look at the log file, it's looking for the manual segmentation. It did not find it. So it's gonna rerun the spinal cord segmentation on this subject, right? So this shows you how you can, create a completely reproducible pipeline by, after modifying some segmentations, after manually correcting some segmentations, packaging your manual segmentations. Let's say you can take your derivatives folder, you can zip it, send it to your collaborators, and then you can tell them, look, you just have to download this data set, install my derivatives, and run the processing, and you should have the same results as I obtained. And it's really a nice way to make sure that your data is your analysis pipeline is reproducible. Any question?
Yes, maybe one. Why do we have to rerun the entire scripts and not just what we have changed?
That's a great question. Because we are wasting a lot of resource here, right? Let's say that you want to you're doing your QC in like July, and then you go to on like holidays for like 2 months, and then you submit your paper, and then you get your reviews, and the reviewer asks you to rerun the processing with one different command. And in the meantime, SCT was updated from 6.5 to 7.0. And then you rerun only the subject that has changed on the one that you modified the manual segmentation. And then you'll have one part of the subjects processed with SCT 7.0 and the other part with SCT 6.5. And that's a problem, right? So rerunning the entire pipeline on the whole data set using a single, like software ecosystem is a way to ensure that all your subjects have been processed using the same scripts reliably. That being said, if you're organized and you trust your organization, then of course you're free to ignore that, to make your scripts more complex, to have some if cases and so on. So that's up to you.
Okay, thanks.
Another argument is that if you want your colleague to reproduce your analysis, then your colleague will have not run those, the first pass of analysis, right? And then if you have a script that says, run, like, don't run this because I've already run that, then your colleague won't have those results. So that's going to be a problem, right?
Yeah, I get it.
I've been doing image analysis for 15 years, and I've encountered those cases hundreds of times. And it's a waste of time. And it's, so just to make sure that we don't run into this thing, that we don't waste time, I'm simplifying pipelines. I'm just saying, run this, it's gonna work. All the subjects are processed with the same script, period.
Okay, that's good for me.
Yeah, we are running a little bit out of time, so I'm going to skip that part. Okay, so we are now talking about the new features of SCT, new and upcoming features. The new features are really revolving around the SCT deep seg function. So we have a couple of new models that have been introduced. One of the model is one to segment spinal cord injuries and then compressed lesions in case of traumatic spinal cord injury. And I have almost no more voice, so I'm going to give my mic to Jan, who is going to take over.
Yeah, so hi everyone. So yeah, as mentioned by Gillian, so here we have a recently developed deep learning method for segmentation of intramedullary lesions in spinal cord injury. Yeah, the method is called SCI-SEG, spinal cord injury segmentation. And it was developed on T2-weighted images with different field of use. So meaning we included images from cervical, thoracic, and lumbar spine. And the images came from different centers with different resolution and orientation, such as isotropic, axial, sagittal. So the method generalized pretty well. And it generalized across different image parameters, but also across different spinal cord injury etiologies, because we can have traumatic spinal cord injury, we can have ischemic spinal cord injury, and we can have also non-traumatic spinal cord injury, which is called degenerative cervical myelopathy. And as you can see in this figure, spinal cord injury patients can have like different phases, because if you are injured, then like your lesion is evolving in time. So you can have like acute patient, intermediate patient, chronic patient, and you can see how the lesions are changing their appearance across these phases. So we developed this algorithm to be as generalized as possible. So here is an illustration how it works. So it first segments the spinal cord, and then it segments the lesions. I believe we can move on to the next slide. Yeah, here we have an example on how you can apply this model SCI-SEG to your data. You can either, we are providing an image with a fake lesion. Why fake lesion? Because it's not easy to share patients' data, but if you have any patients with degenerative cervical myelopathy or spinal cord injury, you can try the method on your data using SCT deepseg command. So you need to specify the input image, and then you need to specify that you want to run this model, this method. So you need to specify task, seg, SC lesion, T2-weighted SCI. And here is how it looks like. So input image, and after running the algorithm, you get your spinal cord and lesion segmentation. Yeah, that's it.
Thank you, Jan. And there was a work in collaboration with Naga Kartik as well. The other model that is being actively developed is the one to segment MS lesions across multiple domains. And that's a project that is being mostly developed by Pierre-Louis Benveniste. And there is also another model for MP2Reg, specifically for MP2Reg, developed by Niels Serre. So very briefly, this model is callable using the task seg-ms lesion. It has been trained on data from about 3,000 patients from at least 20 different institutions worldwide. We've validated it against data from outside institutions, so institutions that were not used to train the data, including data from you, Chris, from UMass. You're gonna see that at the names workshop at ACTRIMS coming up. But yeah, this is very exciting, and we are currently cross-validating it across different other data sets and fine-tuning that model. But it's coming up. Actually, this is already available in 6.5, but we are, we continuously validate and improve that model. There's also an improved version of lesion volumetric analysis, and Jan is going to present that.
Yes, so once you have your lesion and spinal cord segmentation mask obtained using the algorithms mentioned on the previous slides, you can use these segmentation masks to compute various morphometric measures of the lesions, such as number of lesion, lesion length, lesion volume, or also, for example, tissue bridges. Maybe I can just say a few words about tissue bridges because tissue bridges represent a new biomarker recently added to the spinal cord toolbox. This biomarker is popular in spinal cord injury, and it quantifies the amount of the spared tissue in patients with spinal cord injury, and it's illustrated on the figure on the right side. So here you have the lesion. The black guy is the spinal cord, and here we have the tissue bridges, ventral and dorsal tissue bridges. So tissue bridges quantify the amount of the spared tissue. And if we move on to the next slide, we have a syntax which we can use to compute all these various measures. So we can use SCT analyze lesion function, and this function requires two inputs: the lesion segmentation mask and the spinal cord segmentation mask. So basically here we have the inputs, our segmentations, and once we run the command, you will get the output in a form of Excel file. And you can see that this Excel file contains the number of rows corresponding to the number of lesions. So in this particular example, we have a single lesion, so we have only a single row. If we had multiple lesions, we would have one row per lesion. And one recently implemented new feature is a new QC report for tissue bridges. So if you are interested in quantifying tissue bridges, you can also use this QC flag for SCT analyze lesion function, and then you will get something like this. You will get a QC report quantifying tissue bridges for your lesions. And moving on to the next slide, the SCT analyze lesion function also allows you to provide a folder containing the template and atlas registered to your anatomical image. And if you specify such a folder using the flag F, then the SCT analyze lesion function computes the proportion of lesion within each vertebral level and each region of the template and atlas. So for example, you will be able to quantify the amount of the gray matter or white matter or individual white matter tracks occupied by the lesions. So you will, for example, see that 40% of the corticospinal tracks are influenced by the lesion. So again, here is example syntax. So the command is the same as before. You just need to include this extra flag. And since this function relies on the atlas template registered to your image, you might need to run SCT warp template command before if you didn't run it. Yeah, that's it.
Thank you, Jan. Next new feature is the gradient echo EPI cord segmentation. So I showed you in this course that we were using the contrast agnostic model to segment the spinal cord. But in some cases, in some, bad quality EPI, gradient echo EPI, that is, that is difficult to obtain in a reliable way across the population. A student, Rohan Banerjee, developed in collaboration with Merve Kaptan, developed a new model for segmenting EPI, gradient echo EPIs, and that is really that is fairly robust across multiple sites. So the data, the model was trained from a collection of data from multiple sites. Those data have also been uploaded to OpenNeuro, so they are like in open access. And then there was some cross-validation by training on one particular set of sites and then testing on the other sites and so on. So this slide here shows the quality of the segmentation, comparing PropSeg, which is one of the first segmentation tool in SCT, and then DeepSeg SC, contrast agnostic, and then the new EPI seg method. So give it a try if the contrast agnostic does not work well for you, and give us some feedback. We also have a new tool for segmenting the spinal canal, which is the spinal cord and the CSF space that was developed by Abel, who is, I think, on the call, Abel Salmona, yes. Following up on the work of William Sirois. And that tool could potentially be used to then extract some measures of maximum canal compression, for example, in the case of spinal cord, traumatic and non-traumatic spinal cord compression. We could find other applications also if we want to compute the relative atrophy of the spinal cord by comparing the cross-sectional area of the cord over the cross-sectional area of the spinal canal. So you can try this model already on the V6.5 by running SCT DeepSeg, your image, and then task canal T2-weighted. It's still, under development, so please try it and give us some feedback. We would love to see how it works on your data. Another feature is full segmentation of the spine. I'm not talking about the central nervous system here. We're talking about the spine. Nathan, would you like to say a few words? Because that's your baby.
Yes. Maybe you have already tried SCT, especially the function SCT label vertebrae that was presented, I think, the last course, yesterday actually. But the thing with the method is that it's not reliable on some contrast. So the idea was to use deep learning to improve that and extend this ability to locate the different disc and vertebrae on MRI scans. So this method is pretty new, and it's also segmenting the spine, so the individual vertebrae and disc. And at some point, I think one good addition to the model would be to also extract potential metrics from the size of the disc and the vertebrae. But this is not already the case, but this is for future investigation. But yes, if you have maybe time to try it and provide some feedback about the tool, yeah, I think it can be really interesting for a lot of you. So yes, it's also inside the SCT DeepSeg. So so feel free to test it and give us some feedback.
Thank you very much, Nathan. Yeah, so you have all the comments here. I'm gonna address one of the questions from Chris here about the so there are 2 points. Well, actually, the point is really about processing time. So processing time is becoming more of an issue recently with the recent deep learning models, which are heavier than before. I could say two things. One is, so Chris, you mentioned about, if that helps, if you crop the image, yes, it will definitely help. The smaller the image, the faster the processing. That's one. And two, we also offer a GPU installation of SCT to run the inference on GPUs. If your hardware supports CUDA GPUs, like typically NVIDIA GPUs. I don't know, I forgot if we have that covered in the slides. I don't think we do. But we will add, maybe Mathieu or someone else can add as a reply to your comment, Chris, the link to how to do the GPU installation SCT. It's currently an internal feature because we want to test it internally before, making it out large. But since you ask, we would be pleased to have you as our alpha tester.
I run my analyses on a Mac as well, but it's an ARM-based architecture, so it causes all sorts of problems in general. So, but I was just curious, what is your average sort of ballpark CPU time for the MS segmentation pipeline in general? Is it hours and hours, or do you find that it's typically closer?
It depends on the number of subjects, but I mean, if the image is not too big, I mean, usually like less than 3 minutes per subject. I think is what we
okay.
get.
I'll try cropping. Something is going wrong then. Thanks,
Yeah.
John. Appreciate it.
Try cropping and then let us know. Maybe you have some interpolation going on in your images. Maybe they're like, 0.25, because depending on the vendor, they run some interpolation. So you might want to reinterpolate before running the. But yeah, let us know on the forum or via email, and then we'll definitely assist you there.
Okay. Thanks, John.
Spinal nerve rootlets segmentation. By the way, I noticed that we are running a little bit behind. Apologize for that. For those who can stick around, we still have, about a bit more than 10 slides before the end of the course. So bear with us, and you could also see the result of the poll, which could have been interesting. Sandrine, would you like? Oh, sorry, Jan on the nerve rootlets. Please go ahead.
Yeah, sure. So one more deep learning model recently introduced into SCT, and this one is for segmentation of dorsal cervical nerve rootlets. So nerve rootlets are these tiny structures coming out from the spinal cord, and they appear as these small black lines on T2-weighted images. So we developed a deep learning model to segment them. And why, you could ask why we are interested in segmentation of nerve rootlets. So it was partially answered in the previous sections, because nerve rootlets can be used to estimate or obtain spinal levels. So the picture on the right shows spinal levels together with vertebral levels. And you can see that, for example, spinal level C8 is located slightly above the vertebra C7. And there is some variability between subjects. So especially in group analysis, especially for people working with fMRI, it might be relevant to use spinal levels obtained from the nerve rootlets for your registration to the template. And I believe this is going to be covered by Sandrine in the following slide. So if we move on to the next slide, we have an example command how we can use the model for the segmentation of nerve rootlets. So again, it's part of SCT DeepSeg, and you will specify this task. And the output is level-specific segmentation of nerve rootlets. What do I mean by level-specific? It means that nerve rootlets are segmented separately for each spinal level. So you get nerve rootlets for level C2, for level C3, C4, and so on. Yeah, so this is a recently introduced feature, so please feel free to try it and get some feedback.
Thank you very much, Jan. Moving on to the upcoming features, so the ones that are not yet implemented, but soon to be. And we're going to start with Sandrine.
Yes, so Jan introduced it perfectly. What can we do with rootlet segmentation? So concretely, for template registration during the course, we use the vertebrae to align the subject to the PAM50 template. However, we know that the vertebral levels do not correspond exactly to the spinal levels, and also it may differ across subject. So this is very limiting for fMRI group analysis, for example, that we require a good alignment across subject. So here, I am using the automatic rootlet segmentation that Jan just presented to improve the template registration. So more specifically, first, we used the spinal cord segmentation as we did, but instead of having the vertebral levels now, we use the rootlet segmentation. And we also provide the rootlet segmentation in the PAM50 template. And we align each level of rootlets with the PAM50. And with that, then we add the normal steps that we usually do in the PAM50 registration, so the XY. So the main difference is instead of aligning the vertebral levels, we align directly the rootlets from the subject space to the PAM50. And so we would get then our new registered image. And if we go to the next slide, this is a comparison of on the left, we see this is the PAM50 template, and we see the ventral rootlets. Here, I ran the pipeline on the spine generic data sets on 233 subjects, and I created an average of that image when we use the rootlets to register to the template. And we can very clearly see the alignment of the rootlets here. But when I use the discs, so all the cervical disc for the alignment, as we saw previously, it's very difficult to see the rootlets when we average across the subject. So this will soon be included in SCT, and we're very excited to try it and test it also on fMRI data to see if it improves.
Thank you, Sandrine.
That's it.
Very excited about this figure. I think it's a candidate for a cover page of a journal. All right. Let's move on to the poll results, going to the end of the course. What are your answers if you're interested in looking at that? OS, okay. So pretty much the same Windows, Linux and Mac, and then fewer users of Windows. That's very helpful for us. SCT, structural morphometry, microstructural MRI. Okay, so functional MRI coming up, but still the majority would be morphometry and microstructure. Good to know. Feel free to, by the way, this is, we are like, and we enter the discussion stage now, so feel free to interrupt, tell us some comments, feedback. Just going to continue going through the slides, but feel free to interrupt. Specialize, I can see it's still popular. ITK Snap as well. Okay, very few users of 3D Slicer. I'm a bit surprised seeing that, but okay. Right. So, okay, I'm not surprised either. I have to say that even though this is a feature, I kind of discouraged you to use the plugin by showing you the batch processing pipelines, right? Because if you do batch processing, then you should not have to run your processing using the Facialize plugin, right? This is really like a different approach. So personally, I like to do batch processing, but if you're way more comfortable using Facialize, interface, then that's also definitely good. Okay. What is that? What do you use your automates? I see. Okay, well, I'm gonna look into that more details. That's a little bit confusing. I'm not sure what to make out of that. Oh yeah, because we had an other section. I see. So a lot of people not other. That's why. Bids, I strongly encourage you to start organizing your data according to bids. It's going to benefit you in the long term. It's a bit of a pain in the beginning, but then you're gonna find it helpful in the long term, especially if you run a lab. Okay, that's also useful. 69% out of 23 responses. All right, well, thanks for participating to this poll. This is very helpful for us. I'd like to spend, the last 3 minutes just concluding this this course. If you need help related to SCT, of course, I cannot provide help outside of SCT, depending on the case. But you can run the help command to get more information about the input parameters. We did not go through all the parameters during the course. It was not possible, but you might find useful flags for your particular situation. If you're not sure what to use, what command to use, what function, what parameters, you're unsure if your image has too many artifacts and so on, feel free to post on the forum. You see that it's a very active forum. Response time is, usually, less than 2 hours. And, always happy to see that people, give some feedback, and that forum is also, now a useful resource for you as well. If you use the search module here, you might already get some answers for your particular problem. If you would like to be added to the mailing list to be informed of the future courses, you can do so at this address. How many are we? 46. Yes, let's do a Zoom picture before you all leave. This is, traditional thing. We all do that now with the virtual course. It helps to show our sponsors that there is a good attendance. So please switch on your camera if you wish so, and let's take a nice Zoom picture. I'm just gonna move this thing here. Okay. Ready? 3, 2, 1. And then I'm gonna do the other because I have 2 pages. So smile again. 3, 2, 1. Awesome. Thank you so much. And if you, by the way, if you really don't want, because these slides are gonna be public, so if you really don't want to have your picture out there, let us know, and we'll remove it. We'll make sure to remove it. What else is there? Contribute. Yes, it's an open source project, so by definition, we welcome any contributions. From people outside of the lab. It has been the case. We have recent contributions from collaborators in Munich, for example. So if you feel like you have a nice idea, something you'd like to contribute to, please reach out, and we would be happy to assist you. And finally, I'd like to acknowledge everyone who has been contributing to SCT, the core developers, Joshua, Mathieu, Yann, Naga, Nilser, Pierre-Louis, Nathan, Sandrine, Rohan, Yehuda, Abel, and also the previous contributors who left the lab already. I'd like also to acknowledge the sponsors who contributed financially to support this event and the preparation of this event. And obviously, thank you, all the SCT users, for your feedback, and for using SCT and citing us. It really helps to sustain this open source project, getting grants, and paying all the different salaries to make sure that this project is ongoing. Any discussion, comments from anyone? We can actually stop the recording.